everyone. I hope you all have a fantastic evening and are ready for another inspiring and engaging day here at National Conference on the theme Artificial Intelligence on Drug Discovery and Development. Yesterday was truly incredible. We learned so much from our brilliant speakers and had some thought-provoking discussions and made some great connections. And today we have even more exciting sessions lined up for you. Before we dive into today's program, we would like to thank all of you for your enthusiasm and participation, as well as our esteemed guests, faculty members, and of course the organizing team for making this conference possible. Without further ado, let's kickstart day 2 of the conference. Please join us in showing respect as we reflect on our shared values and pride. Let us now call the choir for the Tamil Thai Varthu. Ladies and gentlemen, may I kindly ask everyone to please rise as we honor and celebrate our Founders' Day. Let us stand together in recognition of the remarkable legacy of the Dr. A.M.K. Jambulingam Mudaliar sir.
இரண்டு நிமிடம் அனைவரும் மௌன அஞ்சலி செலுத்துமாறு கேட்டுக்கொள்கிறோம். Thank you. Please be seated. We are deeply honored to announce a special tribute to Dr. A.M.K. Jambulingam Mudaliar, the visionary who laid the Sastha Pharmacy Group of Institutions, whose leadership, dedication, and values continue to inspire us every day. During this event, we will reflect the contribution he made in building this empire. I kindly request our chairman, Dr. Professor J. Karthikeyan, Dr. V.DANIEL, Principal Sir, to commendate our dearest founder. And now I request our faculty members and students for a floral tribute to our founder.
Thank you, everyone. His insights are incredibly valuable to the pharmacy community, especially for our students and faculties. He is none other than our Honorable Principal, Dr. V.DANIEL, sir. We would like to take your time for delivering the welcome address. Over to you, sir.
Coming together is the beginning. Keeping together is progress. Working together is success. Edward Everett Hallett. A very good morning to one and all present here. With warm greetings and give me a great pleasure to extend warm welcome to our beloved chairman sir and our eminent speaker, Dr. Balaji Ramachandran, Professor, Cancer Institute, Adayar, Chennai, and other participants including students and faculty from various colleges throughout India. I would like to thank our Almighty to give a chance to stand in front of you. Our institution is running by A.M. Kaniyappa Mudaliar and A.M. K. Jambalingam Mudaliar Educational Trust. Our trust is started by Dr. A.M. K. Jambalingam Mudaliar, that is the founder chairman. Today is the founder's day. We remembered our founder chairman. He was one of the trustees to start Vellore Institute of Technology in Vellore. He only the person to give the whole finance to start the Vellore Institute of Engineering. At the time, it is a Vellore Institute of Engineering. Nowadays, it is called as Vellore Institute of Technology. The three persons started it. First, the Vellore Institute of Technology. One is the Jambalingam Mudaliar and G. Vishwanathan, and one other is the Kumaravel. The three persons started the Vellore Institute of Technology. Our founder chairman is a finance, the treasurer, given the whole finance to start that institution. Now this institution is the #1 institute in India in engineering technology. After few years, he relieved from the Institute of Engineering Technology. He was started the Sree Sastha group of institution in 1998. Nearly 26 years gone. He started lot of colleges, engineering, Institute of Engineering Technology, Sree Sastha Engineering, Sastha Education College, Sastha Nursing College. After he expired, his son, now a chairman of Professor J. Karthikeyan, he started the Pharmacy College, an Architecture, Arts and Science College. In few days, we will start the Paramedical Science College. Already inspections over. We will start the new course, Physiotherapy as well as the DMLT very soon. So our institution nowadays, the Professor J. Karthikeyan B. MBA. He completed the B is the Manipal, MBA in London. He is an educationalist. He has deeply committed to the responsibility of training and the students and staff in terms of attitude, knowledge and skill sets to face the real practical life and professional life. I would like to thank our chairman sir to permit to conduct this national conference in our college. I would like to thank the Vice Chancellor, Tamil Nadu Dr. M.G. Medical University, and Dean of the Students, Dr. Sudha Madam, Tamil Nadu Dr. M.G. Medical University, to accept our request and give an accreditation in this conference and allotted 15 credit points with sponsorship with the Tamil Nadu Dr. M.G. University. The Tamil Nadu Dr. M.G. University sponsored national conference is being organized from 26/9/2024 to 28/9/2024 at Sree Sastha Pharmacy College, Chennai, Tamil Nadu. Eminent speakers delivered by yesterday Dr. S. Kavimani, Professor, Mother Teresa Institute of PG and Research Institute of Health Sciences, Puducherry. He was the member of PCI chairman for the B.Pharm syllabus committee. Yesterday, the eminent speaker delivered a very good speech, Dr. K. Gilango, Principal, Thakur College of Pharmacy, Chennai. Today, our chief guest and the eminent speaker, Dr. Balaji Ramachandran, Professor, Cancer Institute, Adayar, Chennai. Today afternoon, another one very good experienced and well-known person, Dr. Ubayatullah, Professor, Crescent College of Pharmacy, will deliver the speech. Tomorrow, Dr. R. Vijayabharati, Professor, College of Pharmacy, Madras Medical College, and Dr. N. Kandapan, Professor, Annamalai University, Annamalai, will deliver a very good speech regarding our theme. Theme of the national conference is accelerating pharmaceutical innovation, artificial intelligence in drug discovery and development. So, yesterday two eminent speakers delivered a very good speech. Today we will expect another two speakers. Tomorrow we will expect another two speakers will deliver the very good speech according to our theme. So, as a pharmacy people, we must know about the artificial intelligence. Artificial intelligence is very good. Nowadays it is one of the very developed departments. Another one is the whole world to be occupied by the artificial intelligence. So artificial intelligence is a stream of science related to intelligent machine learning, mainly intelligent computer program, which provides result in the similar way to human attention process. So role of artificial intelligence in pharma and healthcare: automated quality control, better compliance, cancer research, drug discovery, drug repurposing, medical occurrence, personalized treatment, predictive forecasting, as well as the streamlined production. That is, introducing artificial intelligence into the drug manufacturing allows to firms and keep the production lines running. This technology helps to predict and address potential breakdown of the supply chain and malfunction with the manufacturing tools. The another one disadvantage for the artificial intelligence: the human job will be reduced in future. Only machine do all the works automatically. The human job will be reduced. That is the only disadvantage and main disadvantage for the artificial intelligence. But we must know what is artificial intelligence, how to rectify the problem, how to solve the problem. So without knowing artificial intelligence, we will not be able to understand. First we understand what is meant by AI, then how to solve the problems to lose the job opportunities. With the human being, with the human power, the artificial intelligence is essential. Without human power, artificial intelligence will not accept it. So first of all, we must know about the artificial intelligence, how to neglect, how to use. So in future, we will understand. This time I give the thanks to all the participants and their various colleges, particularly yesterday nearly 20 colleges from outside college peoples participate this conference. GRT College of Pharmacy, Apollo College of Pharmacy, GRD College of Pharmacy, Balaji College of Pharmacy, Bharat College of Pharmacy, CL Petwega College of Pharmacy, Satyabama Institute of Science and Technology, Chettinad School of Pharmaceutical Science, SRM University, St. Peter's Pharmacy College, Hosur, Sri Indira Ganesh College of Pharmacy, Trichy, Hindustan Institute of Science and Technology, Savita College of Pharmacy, Wales College of Pharmacy, Sri Ramachandra Medical College and Research Institute, Sri Sastha Institute of Engineering Technology, Meenakshi College of Pharmacy, Tandai Rover College of Pharmacy, Muhammad Sadek AJ College of Pharmacy, Cogent School of Business and Technology. So there are 20 college students participate this conference. Yesterday conference, today's conference, we will expect tomorrow they will attend this conference. I thank all the principal, particularly GRT College of Principal, Dr. Meenaji Sundaram, Apollo College Principal Satyanandan, GRT College of Principal, Dr. Sadisha, Balaji College of Principal, Dr. Deepa Madam, Bharat College of Pharmacy, Dr. R. Srinivasan, CL Pate Mehta College of Pharmacy, Dr. Nalini Madam, Satyabama Institute of Pharmacy College, Science and Technologies, Sanmuga Padil sir, Settinad School of Pharmacy, Dr. Lakshmi Madam, SR University, Dr. Chitra Madam, St. Peter's Pharmacy College, Huzur, Dr. Premanand, Sri Indira Ganeshan College of Pharmacy, Dr. Nirmala, Hindustan Institute of Science and College of Pharmacy, Dr. Surya Prakash, Savita College of Pharmacy, Dr. Ray Murugan, Wales College of Pharmacy, that is the Director, Sanmu Sundaram, SRMC, Dr. Jarrah Suresh sir, Sri Sastha Engineering College, Dr. Sanmu Sundaram sir, Meenakshi College of Pharmacy, Dr. Somaneshwari Madam, Sanday Roja College of Pharmacy, Dr. Napoleon sir, AJ College of Pharmacy, Dr. Sundarajan sir, Ozil School of Science and Business Technology. I didn't know it is not considered pharmacy, so I didn't know who is the principal. So thanks for all the principals who allowed your students for this national conference. Thank you for all the participants. So we hope another 2 days you will get more information about the artificial intelligence. Thank you one and all. God bless you one and all.
Now we call upon our beloved principal sir to felicitate a chief guest on behalf of our institution.
At this time, I would like to call upon our beloved staff, Mrs. Aparna. She has been a key figure in organizing this conference and we are pleased to have her with us in behalf. To address our speaker, Dr. Balaji Ramachandran. Over to you, ma'am.
Good morning to everyone who attended the conference today. Before I start giving the introduction regarding the speaker, I would like to give a quotation regarding him based on his researches. Unlocking the secrets of cancer biology to develop innovative, effective and targeted therapies in the holy grail of our researchers' endeavors. Dr. Balaji Ramachandran is a renowned professor at the Cancer Institution in Adyar, Chennai, with a strong background in pharmacotoxicology and pharmacological biotechnology. He had made significant contributions to the field of cancer research through his extensive experience in animal models, molecular imaging, and in vivo pharmacology. With an illustrious career spanning over 14 years, Dr. Balaji Ramachandran has established him as a leading authority in pharmacotoxicology and pharmacological biotechnology. With a strong educational foundation in pharmacy from the Tamil Nadu Dr. MGR Medical University and biotechnology, MSc Biotech from Alakappa University, Karaikudi, and holding a PhD in pharmacological sciences from the University of Milan, Italy. Dr. Balaji Ramachandran has been an instrumental in shaping the institutional research agenda and maintaining the next generation of cancer researchers and clinicians. His innovative approaches to cancer therapy have earned him national and international recognition. Dr. Balaji Ramachandran has published articles in esteemed journals with a total JPR impact factor of 89.42 as a scientist in charge of Animal Facility and Member Secretary of Institutional Animal Ethical Committee since 2010. And he has received several prestigious awards and honors for his contribution to cancer therapy. It is been as a reviewer in Oncology Reports, International Journal of Endocrinology, Oncology Letters, Gene and Medical Oncology. His research expertise in cancer biology, drug discovery and translational research, and his research focuses on cancer pharmacology and toxicology, drug discovery and development, translational research and precision medicine, cancer stem cell biology, nanotechnology based on cancer therapies. Dr. Balaji Ramachandran's vision is to develop innovative, effective and target cancer therapies that improve patients' outcomes. His mission is to advance cancer research and education through collaborative efforts fostering a new generation of cancer researchers and clinicals. As a professor at the prestigious Cancer Institution, Aliyar, Chennai, Dr. Balaji Ramachandran continues to inspire and mentor the next generation of cancer researchers and clinical. Now it's our pleasure to have Dr. Balaji Ramachandran, Professor, Cancer Institution for today's session's keynote addressing on in vivo imaging as a non-invasive screening tool in transitional research. We are honored to have you to share your insights with us today, sir, so which will enlighten our younger generation. Thank you, sir.
We are obliged to call our esteemed speaker on behalf of our management to share a few words. The floor is yours, sir.
So Good morning, one and all, and I thank the management and my senior, Mr. Shyam Sundar, for giving me this opportunity to be here. I'm Balaji, and I'm basically a pharmacist, and then next is that I'm a pharmacologist. So, like, after completing my B Pharmacy from Dr. LGR Medical University, I did MSc Biotechnology. I didn't do M Pharma. So after that, after the master's degree, I went to Europe to pursue a PhD degree. So there I learned something called in vivo imaging, so which is very translational kind of subject, and which is also a state-of-art subject, which the data that is coming out of that subject is something like where the artificial intelligence could have some a big role. So like Dr. Dhanapal said, like when he was speaking, he said that due to AI, humans may lose job, but this is basically an animal job, and the data that you get out of it needs to be analyzed by some humans, and that too, AI would act as an interface to it. So before going into my presentation, so sir asked me to speak for just 5 minutes. So I thought that I would better talk about my department, so that you are young guys, like you are in the bachelor's. So you can have an understanding like, what would be your future prospects after completing your B Pharma. It is always not necessary like you have to be like, after D Pharma you have to go for M Pharma and then PhD or something like that, join Sutix, do Sutix or go for pharmacology, something like that. It's not like that always. Like you can always do, there is other stream where like, you can, there is also an option to a master's in science or a master's in technology, and then you can come back and like you can still stay in the field again. You can be back to the form field. So like after completing my PhD, so I immediately came back to India. So there I was headed by a clinician. So I was like associated with a panel of doctors. So they are basically like clinicians, and they have a clinical knowledge only. So like we have there is another set of faculties including me, so who are basic or translational scientists. So we always work together, and that has formed a kind of interface, because like clinical knowledge when you have to do something in the bench, and when you want to translate it, you can go to the clinic and do. So our department, molecular oncology department in Cancer Institute. So where I am associated with the institute through this department for the last 15 to 16 years. So this is what we are all been doing in our department. So there are so many phase 1 and phase 2 clinical trials that have come up from the department, and the department was started in the year 1996, and now we are in 2024. So we have done so many firsts, and we have done a lot of technology transfers kind of thing, and there is a good opportunity for people who have done D Pharm. I think it's a 5-year course or 6-year course. I've seen a lot of people working on clinical trials in our institute. So 6 years. So there are good opportunities. It is not that necessary that you always have to keep a pharmacy or something like that after completing your B Pharma. There are very good opportunities for you waiting, and I think the topic AI is like, I'm little delineated from that topic, but in order to give you the data for AI, you need to do some groundwork. And coming to our field again, nothing is possible without human intervention. So like you have a drug or you have a compound or something in your hand, it doesn't mean that like without testing by humans in some models, it cannot go directly towards therapy. So we cannot take any risk in treating something directly to humans. Like we always have to do something in the interface, and that's all about that I'm going to talk about. And the data that would be generated in case if you want to do a high throughput screening, such data would work as a kind of a raw material for your artificial intelligence application. So with this I conclude, and I think we can go for a break or something if you agree. So maybe later on we can continue with our slides. Thank you.
I would like to remind the oral presenters to kindly ensure to have their files in a USB drive to share your contents at the pharmacy block AV room 3rd floor. connection cables or email to sspccon2024@gmail.com. I repeat, sspccon2024@gmail.com. Apologies, it is sspccon2022@gmail.com. This will help us to smoothly connect to the system and avoid any delay while it's time for your presentation. Thank you. And we will now take a short break. This break will last for 15 minutes. Please feel free to stretch, grab some refreshments and return to your seats by time. Make sure to adjust the duration. The break is for 10 minutes. Please feel free to stretch and return to your seats by time. Make sure to adjust the duration and time based on the actual schedule, as we will resume with our next session promptly. Enjoy your break and we will look forward to continue the conference. USB drive at pharmacy, 3rd floor. Restroom?
Kindly please use the restroom at ground floor. Ground floor and 3rd floor.
மேலே 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 மேல் ரோ.
Hello, check.
I would like to give again a gentle reminder for the oral presenters that as we prepare to present, kindly ensure to have your presentation file on a USB drive to the pharmacy block AV room 3rd floor. And you can also use your connection cables or email to the ID Sspccon 2022@gmail.com so that they can proceed your presentation smoothly. Any queries about the presenters? And those who are presenting are requested to go to the pharmacy block AV room 3rd floor. Welcome back everyone. I hope you all had a refreshing break. We are now ready to start the first session of today's conference. We call upon Dr. Balaji Ramachandran for their presentation. Over to you, sir.
Nara outer please. Sastha. Hey, adilada. அது பார்க்கலாம். நமக்கு லைட் வரும் ஒன்றும் பிரச்சனை.
We request all to settle down quickly to start the session. Please maintain a silence.
Are you comfortable with English or Tamil or both? Okay, so இங்க எத்தனை பேர் வந்து pharmacist எல்லாருமே pharmacy தான் இல்லையா? So pharmacology வந்து ஒரு வருஷம் மட்டும் தான் இருக்கா? இல்ல advanced pharmacology நாங்க படிக்கும் போது ரெண்டு எத்தனை 3 papers. So basic to next advanced come. So it was like another particular it was like third year, fourth year, starting the basics of the fourth year we had an advanced competition. So, the intention of making this presentation here is that this will be very, very useful for people, for candidates who want to pursue a career in the field of pharmacology. So, whatever I am going to show you now, if you get experience with that techniques and technologies, you will definitely have a good chance to get placed in a pharmaceutical company as a preclinical scientist. So this is basically a pharmacology based topic. So the topic title was in vivo imaging as a non-invasive screening tool in translational research. So translational research na enna theriyum ya. So you do something in the bench, and the bench work எடுத்து போய் நீங்க வந்து clinical application எடுத்து அத வந்து எப்படி அப்ளை பண்றீங்களோ அதுதான் translation. So இது வந்து ஒரு கோர் சப்ஜெக்ட். இந்த கோர் சப்ஜெக்ட்ல வந்து நீங்க வந்து கத்துக்கிறது வந்து பாத்தீங்கன்னா அதுல இருக்கிற அவுட்புட்டை வச்சு you can always go for AI based tools. But before going to AI based tools, you must need some data. So அந்த data வந்து you can get out of this kind of state of technology. So next slide please. So this is about our department, molecular oncology department, with which I am associated for the last 16 years at Cancer Institute WA. So this technology is a state-of-art technology. So this is a very advanced technology. What I am going to talk to you about today. So every technology is being inspired from nature. So like you see, this is how you see the Earth from a space. So there are sometimes it has been reported in newspapers saying that sailors report steaming for hours through the sea glowing. So in such instances, the causal organisms here is there nothing but the dinoflagellates, and so this they call it as a ghost or something like that, but these are actually nothing but the bioluminescence produced by algae and bacteria. And this can extend up to 450 kilometers in the sea. And even sometimes mechanical stress induces bioluminescence. So today's topic is all about bioluminescence imaging and cognitive imaging. So in this, next slide please. So in this presentation, I am going to talk about the basics of bioluminescence and what are the details. I am going to give you the details about bioluminescence imaging. I am going to compare the bioluminescence imaging with other modalities, and we will also see the application of bioluminescence imaging in pharmacology. So what is bioluminescence? Bio means biology, living system, and luminescence means light. So bioluminescence is basically the production and emission of light by a living organism as a result of chemical reaction, and ATP is being involved in most of the instances. So bioluminescence or event what are all the things that you will be needing basically? So you need an enzyme called luciferase, and you need a substrate called luciferase, or it can be any substrate, and of course you need a molecular in vivo imaging equipment. With these three basic things, you will be able to carry forward bioluminescence imaging. Next slide please. So what is basically happening in bioluminescence reaction? So this is basically an oxidation process in which the luciferin, for example, you can take a substrate luciferin, and then it reacts with ATP, it is converted to oxyluciferin and light. So this is basically an oxidation process, and the energy difference will be released as a light, and this light will be very energy efficient and will be captured by a CCD camera. So the imaging equipment part I will come in the later slides. Next slide please. So coming back to the nature, so you can see bioluminescent bacteria in the petri plate, you can see the protozoa, the fungi, the insects, even the glowworms, they glow and they produce light in nature, even the squid can produce light. So fortunately no mammals produce light. So So that opens up new avenues for applying bioluminescence technology in the field of preclinical testing. So this bioluminescence imaging technology is a preclinical screening tool. Next slide please. So now let's have a quick look into the natural luciferases that are available in the market. So this bioluminescence imaging concept of it was started in the year 1990. So it was in the very beginning level, there is something called Luxopron, which has been very famous at that time. So Luxopron, so as I told you in the previous slide, you need a substrate and an enzyme. So அதுலயே வந்து லூசிஃபெரினுக்கு ஒரு ஜீனும் இருக்கும் and luciferase synthesize பண்றதுக்கு ஒரு ஜீனும் இருக்கும். But unfortunately this Luxopron is a very beginner method, and scientists find it very tough to clone such a huge amount of thing into any transcription system. So then shorter versions of luciferase, which the substrate is deluciferate and it emits a yellow light, and then regular luciferase for which the substrate is present and it can emit blue light, and there is something called vascular luciferase which is from the poppy fall, and for that the substrate is present, and that is the very brightest luciferase known till date. Now the problem with this Gaussian luciferase is that so whenever this Gaussian luciferase is getting expressed, it needs basically disulfide bonds to be formed in a very efficient manner for it to work as an efficient luciferase. So again, scientists find it very tough to express this Gaussian luciferase and form the proper disulfide bond and get converted into a proper functional protein with the proper conformation. So they just dropped it, and very many luciferases have now been dominated in the market. So one such thing is from Tulu, which is a railroad worm. So it basically synthesizes two different luciferases. So in that, one is red, which we can widely use it for in bioimaging because a red light has minimum absorption and maximum emission. So that is the credit with having a luciferase with red, which can emit in the red zone of the spectrum, visible spectrum of light. Next slide please. So obviously non-optimal luciferases can be ameliorated by rational design and by molecular evolution. So you can simply modify the codon, the translating thing, so that you can modify your luciferases according to your need, and you can increase the brightness or you can decrease the brightness, but normally people will try to increase the brightness normally. So you can also fix the color from fluorescence, red, or you can use a red shade, something like that, and you can even alter the stability. So when you do increase the stability, you can use it for high screen, but if you decrease the stability, you do time resolution. Every time resolution now. See, you have a model in which your luciferase is expressing, and you want to see how the drug is getting bound to that your target. So when the drug binds to the target, then the target will not be available to generate the luciferase. So immediately what will happen? The entire light will get switched off. So what would happen if that light is still there? Then you will not be able to see whether your drug is binding to your target or not. So something like that. You can do the necessary modifications according to your requirement and according to your scientific question that is compelling you to. Next slide please. So if you look into the web, almost 100 different patented, even more than that, patented luciferases are promoted to the market. So this thing is becoming so popular. So why animals should produce life? Next slide please. So if you can next slide please. Go to the so you can see in the nature, coming back to the nature, the angler fish. So attract the prey, and once the prey gets into the mouth of the fish, it can easily enter it. And firefly bees, they dance and they produce luciferase reaction based light so that they can attract their mates. And for example, the shrimps, they emit a kind of a very luminescent gel to get rid of the predators. Next slide, please. And then why a mice should produce light? It is nothing but to shed light in the life of the black box. So what is a black box? I will come to the next slide. So in the past, there is no functional thing like diabetes insulin secretion diabetes so there is nothing like NIDM, IDM. So in those days people thought that the living body is like a mechanical gear, and when you trigger that mechanical gear, a homeostatic response is being elicited, and that response is basically sets forth the clinical outcome. Next slide, please. So it is now we know very well. So when some trigger happens, say or toxic chemical or nutrition or hormonal infection. So what will happen if you take all these drugs and hormones and nutritions? So there will be a difference in the upregulation and downregulation of the genes, and a lot of changes will happen at the molecular level. So many molecular mechanisms will be altered, and that would be basically lead to the clinical outcome. One such good example is the cancer. So cancer, there is a lot of upregulation, lot of downregulation, lot of changes happen, so that and the carcinogenesis event they encourage pandradanaala, so there is a clinical outcome which is nothing but a cancer. So something like that. So next slide, please. So ஒரு pharmacologist என்ன பண்ணணும்னா you have to identify and follow the biomarkers that could predict and allow for monitoring of early onset of clinical outcomes. So in the clinical outcomes எதனால வருது? So அந்த molecular mechanisms எப்படி நம்ம அதை ஐடென்டிஃபை பண்ணணும், எப்படி அதை ட்ராக் பண்ணா இந்த clinical outcome நம்ம தடுக்கலாம். ஒரு கேன்சர் ஒரு எக்ஸாம்பிள். ஸோ எனக்கு இந்த ஜீன்ஸ்லாம் எக்ஸ்பிரஸ் ஆனால் இந்த மாலிகுலர் மெக்கானிசம்ஸ் ஆல்டர் ஆச்சுன்னா எனக்கு கேன்சர் வரும்னா அந்த மாலிகுலர் மெக்கானிசம்ஸ் எகைன் ஒரு பர்டிகுலர் சப்ஜெக்ட் ஆஃப் கேன்சர் எடுத்தீங்கன்னா there are various subsets and each patient is a unique patient. It's a very heterogeneous disease. ஸோ அப்படி இருக்கும்போது அதுக்கான காமன் மாலிகுலர் மெக்கானிசம்ஸ் என்ன, அந்த சப்செட் ஆஃப் கேன்சரில் இப்போ நீங்கள் பிரஸ்ட் கேன்சரில் பார்த்தீங்கன்னா TNBC இருக்கு, ட்ரிபிள் நெகட்டிவ் பிளட் பிரஸ்ட் கேன்சர் இருக்கு, HER2 positivity இருக்கு, ER positive, ER negative, PR positive, ER positive, அந்த மாதிரி various categories, various classifications இருக்கு. So how do you identify the molecular mechanisms of the research topic that you are interested in? So அது எப்படி கண்டுபிடிப்பீங்க? So இப்போ என்ன நடக்குதுன்னா, so like in the hormones, drugs and nutrition, and they all kind of they attack the gene expression profile. I mean, they alter the gene expression profile, which triggers to clinical outcome. Next slide, please. So, நமக்கு பேசிக்கா தெரியும் இந்த இடத்துல ஒரு மார்க்கர் வச்சா என்ன நடக்கும், அந்த இடத்துல ஒரு மார்க்கர் இருக்கு, இதுதான் ஒரு பாத்வே. இங்க ஒரு ஹார்மோன் இருக்கு, ஸோ இங்க ஒரு ட்ரக் இருக்கு, ஒரு ஹார்மோன் இருக்கு, நியூட்ரிஷன் இருக்கு. ஸோ இது எப்படி இதை ட்ரிகர் பண்ணி அந்த ஜீனோமை ட்ரிகர் பண்ணி how the clinical outcome has happened. ஸோ இந்த இடத்தை நீங்க எப்படி ட்ராக் பண்ணுவீங்க? You need a marker to track it. So in the past என்ன பண்ணாங்க, again blood. நம்ம ஒரு உடம்பு சரியில்லை ஒரு ஹாஸ்பிட்டல் போறோம்னா first they will extract the blood and see they will isolate the blood and see the traditional profiles, and அப்படி மட்டும் பண்ணா மட்டும் எல்லா டைமும் நீங்க கிளினிக்கல் அவுட்கம் தெரிஞ்சுக்க முடியாது. So if you consider this cell of a living body to be the key unit of a black box, so you first have to identify the marker. You have to put in some markers to understand what molecular mechanisms are actually altered to get the clinical outcome. Next slide, please. So how to put a marker? So ஒரு gene follow பண்ணனும்னா what you can do, you can put a marker in front of the gene, or you can put a surrogate marker in such a situation that when this surrogate marker, when it gets upregulated or downregulated, when it gets expressed or when it got activated, it will mirror your molecular mechanism of interest. So that you can easily identify the thing beyond, I mean the reason behind the clinical outcome. Next slide, please. So This is basically an instrument, a very prototype of an in vivo imaging equipment, so which was created in the year almost close to 1990s. So you need few things which I will tell you now. So you need a mice and you have to anesthetize the mice. So mammal mice is the best model which can be as closer to a human-like situation. So we cannot put a human inside, but we can use an animal which is especially mice, which is almost closer to a human-like situation. So you can put that animal under a CCTV camera and you can do imaging provided this animal is transgenic animal. So what is a transgenic animal? That I will come to the next slide. So that you can do make a survey of which body districts have been affected by your drug treatment. Next slide, please. So how to study gene expression in vivo? So you can basically study the gene expression in vivo by the method of transgenesis. So it can be a whole animal reporter mice. So you can have an example of a mouse model, which I will show in the future slides. So on the mouse model, each and every cell there will be a reporter gene, so that when you image the mice under the that imaging system, you can understand where exactly this molecular mechanism is getting activated, where exactly your gene of interest is getting altered or activated or downregulated. So to do that, you need a transgenic animal, or it is always not necessary that you need a transgenic animal. It can be a CDX, a cell line derived reported xenograft. So we are working hardcore. Our hardcore job is to work with CDX and PDX models. So PDX means patient derived xenograft. So the translational research, it has more value compared to CDX. PDX has more value compared to CDX, and that's a very vast subject. I don't want to go into that. So with the introduction of reporter genes into animal genomes, they provide opportunities to investigate the activity of a given target of your interest in the context of a living organism. And with the aid of imaging technology, the dynamics of transcription of simple or complex proteins can be observed in real time with reference to space and time. So spatial fashion, temporal fashion, and the details which I will give you the future slides. Next slide. So this is nothing but a imaging, in vivo imaging equipment. So in the CCTV camera. So CCD stands for cooled charge coupled device. So it will get cooled up to minus 350 degrees Celsius, and you have to put your anesthetized mice inside, and provided that mice should be a reporter mice. So when you put that mice inside, there are other factors which will interfere with your imaging technology. Say for example, if a bioluminescence imaging there is no need to excite your reporter gene. You simply have to give a substrate, and that substrate will be cleaved by your enzyme, whatever you are expressing, whatever reporter gene that you are put inside the animal, and that will start to glow, and the excitation it has to get emitted. So usually luciferase it will start say 570 to 600 nanometers, it will emit the light. So that light will be captured by the cooled CCTV camera. So நடுவுல நிறைய factors இருக்கு. Say for example, the animal fur will interfere with the light of the scatter protein, and the presence of hemoglobin will again scatter your light. Despite all these hindrances, still your light will be captured and it will be observed for a period of time, say 5 to 10 minutes by the CCTV camera, and you will get a data output something like that, where you can see here. So say for example, you can see the liver that is glowing. So you can see these images without killing the animal, without sacrificing the animal. You can see you can identify the body district of the animal. Say there are few breast nipples which is being activated by your drug X, Y, or Z, something like that. And you can also follow what is the activity in the bone when you look at the tail of the animal. So you can actually look into the spatial pattern of where exactly your drug is getting activated, and where exactly your molecular mechanisms of your interest is changing, is increasing or decreasing. So those details you can learn from this technology. Next slide please. So with this in vivo imaging technology, it is possible to conceive almost you can do whatever you want with this. It's such a versatile technology that you can apply this technology to say starting from cancer or to infectious diseases or something like that. Say like you put a constitutive promoter and you will be able to track the molecular mechanism at the cellular level, in your model. And if you put an inducible reporter, you can follow the transcription activation of that molecular mechanism in response to your drug, and you can even study protein-protein interaction. Say you have two proteins of your interest and you want to study in an in vivo fashion. See, all these are being done in a physiological setup. So our human system is a physiological system. So ஒரு cellular systemல வந்து பாத்தீங்கன்னா ஒரு in vitro cell culture like it again depends on the total environment of your 96 well plate or your cell culture plate, your 6 well plate or 12 well plate or something like that. But here you're dealing with your molecular, you can see the activation of your molecular mechanism in a physiological setup. So that is the main advantage with this technology. So you can easily identify protein-protein interaction. Say for example, you split the reporter gene into two halves. So you just fuse one half with one protein A and another half with another protein B. So when these two proteins they get when they combine together, there happens a protein-protein interaction, and your reporter gene will be complete and it will go and start to cleave the substrate, and it is going to give you the light. Next slide, please. So imaging modalities have unique advantages, and let me give you a small introduction about the comparison between different imaging modalities. Next slide, please. So molecular imaging, there are three types of imaging. One is a direct method, the other one is an indirect method, and the last one is a surrogate method. So optical imaging is an indirect method. Like if you want to understand the dynamics of any particular gene or its outcome, that is protein, you use another protein as an indirect method, as an indirect marker to track the actual function of your activation of your protein of interest. So that is about one optical imaging, where you use either fluorescence or chemiluminescence, or otherwise called as bioluminescence. So bioluminescence in a manner is a chemiluminescence because it is basically a chemical reaction, that is an oxidation process. And next is nuclear imaging. So in nuclear imaging is a very direct method in which you have the some radionuclides which will directly combine with your target, and you can see the real-time functioning or circulation of your target of interest. And the third thing is the surrogate marker. Say like in MRI, say for example, you have a transferrin receptor, and what you can do, the main job of this transferrin receptor is to take something from the plasma and put it inside the cell. So like there are certain iron oxide complexes which will actually combine with a protein called transferrin, and that complex will be engulfed inside a cell by the transferrin receptor. So natural these transferrin receptors are very abundant in cancer, in a tumor mass, or in a if you can actually find a difference if whatever thing that is being engulfed is cancer or not, first of all. And the second thing is that like you can actually send your drug of interest and see whether it has got any therapeutic effect in your cancer cell of interest. So these are three kinds of imaging technologies. Next slide, please. So here I basically wanted to do the comparison between different imaging modalities. So like optical imaging, if you see, it has a decent level of sensitivity, specificity, linearity. Although spatial resolution is very limited, because like you put the animal in only one dimension, in a very fixed two-dimension situation, but the temporal thing is very good, and processability is very simple, and the cost is very, very cheap. So out of the four other technologies, optical imaging is the one which satisfies all the qualities of anatomical relevance, anatomical resolution, physiological resolution, metabolic and molecular resolution. Next slide, please. So this slide basically compares the cost differences between several imaging modalities. So what you want to, this is a very busy slide, but what you can understand from here is that the optical imaging or the bioluminescence imaging is the cheapest of all the other remaining available imaging technologies. Next slide please. So you take any imaging technology where you can apply it in the preclinical situation, say like a MRI for example, MRI imaging of preclinical animal models. So it has got both advantages and disadvantages. Say for example, the MRI has a high spatial resolution, which is the advantage, and low sensitivity is the disadvantage. And then when you come to PET imaging of preclinical animal models, so it will give you very good readouts about the biochemical situation, but it will give you a very poor readout about the anatomical situation. And then coming to CT, ultrasound and optical imaging. In CT, like you have a very good sensitivity, but it has got a very good poor resolution. So that is the problem with CT. And coming to ultrasound, you can do a 3D imaging with ultrasound, but the problem with ultrasound is that like you cannot image the bone using an ultrasound technology. And coming last to the optical imaging technology, it is very easy to work with, but the disadvantage with optical imaging is that like you need a complex animal model with a genetic manipulation being done as a model is very much required. So that is a potential disadvantage of optical imaging. So if you take the imaging modalities, like there is both advantage as well as disadvantage. Like you have to choose like what exactly your research topic will be wanting, and you can actually make a kind of a hybrid. You can use a combination of multimodal approaches as per the requirement of your research question. Next slide please. So coming to the unique features of in vivo imaging. Next slide please. So like the molecular imaging or the optical imaging or the bioluminescent imaging, so they will help you to study the functional process in vivo. Say for example, you have a ligand. For example, there is an estrogen hormone. So this ligand, when we inject, like it gets into the cell and it will bind to the next slide please. Next, okay. So it will bind to the estrogen receptors, and this estrogen receptor here, so it gets dimerized after it binds to estrogen hormone or estrogen hormone, and then it gets inside the nucleus, and once when it gets into the nucleus, it starts translating the downstream response genes. So you can see the lucifer is growing in a cellular plate. Next slide please. So when you inject these cells, you can see the light growing in the animal model. Next slide please. So next slide please. So here I would like to introduce a paradigmatic patented very good reporter mouse. So this mouse is something very special, and it has been made by a professor called Adriana Nagi in the University of Milan, Italy. So this model has a luciferase gene which is under the control of a promoter called thymidine kinase. So this thymidine kinase is again under the control of a promoter, a response element called estrogen response element. So this presence of this estrogen response element make this construct to be very specific to act, to get activated or to get transcribed when any estrogen or estrogen-like material gets into the system. So this entire construct is being dotted by two insulator sequences. So these insulator sequences, they generally prevent this construct of being by being influenced by distant enhancers and silencers. So the readouts would be like when you inject luciferin into this animal, you can put this animal inside the in vivo imaging system. You can do in vivo imaging of the animal, and then you can take out the organs after your human sacrifice, and you can do ex vivo imaging, and you can also do cellular imaging in the same in vivo imaging equipment. On the other hand, this luciferase gene protein is a very strong antigen, so you can use this for immunohistochemistry analysis. And this is since this luciferase is an enzyme as well, you can use it as a screening tool in ex vivo enzymatic assay as well. Next slide please. So collectively, the battery of all these tests together, they will mirror the in vivo imaging of transcriptionally active estrogen receptors in a physiological situation. Next slide, please. So, this is an acute study in which estradiol, the traditional hormone for estrogen receptor, has been injected into this model. So, you can see from hours, starting from 0 hour, the light is slowly increasing, but by 17 hours, you all know, like third year and fourth year pharmacy students will definitely know about pharmacokinetics and pharmacodynamics. So, pharmacokinetics, it will generally start by half an hour and it will end up by 24 hours. So you can see the light going down. So it's a very flexible and very reasonable model where you can understand the data by just looking at it. You don't have to do a big analysis. You can just look at the pictures and you can understand the data. And this can be done in hours, and at the same time this can be done in days also. It means that you can study the activity of a drug in a longitudinal period of time in the same animal without sacrificing or without killing the animal. You can even avoid intra-animal variabilities when you have this technology in place. And of course, you can quantify the activity of any drug or hormone in a spatial fashion. Like you can put mask, quantification mask like this. Like you can put a head area, thymus, liver, something like that, and you can do quantification using the imaging methods. So now we are doing something called, we are in an era of generating a lot of data output, and that data can be fed into an AI kind of a thing, and you can do a high throughput screening if you are working as a, say, preclinical scientist in a pharmaceutical organization. You can even combine the AI tool with this, and you can get rapid readouts with this technology. Next slide, please. And you can even do a physiological validation. So in the before slide, I've deliberately injected the animal with estradiol, but here I've taken some female mouse, and I'm observing them, those cycling females, and I'm also cross-checking with the menstrual phase these animals are in. So for unlike humans who have 20 days of menstrual cycle, mouse have only 4 days of menstrual cycle. So you have proestrus, estrus, metastrus, diestrus, only 4 phases, and there will be a high amount of circulating estrogen only in the phase of proestrus. So you can see the sex organ, the vagina, showing a high photon emission here in proestrus, and liver is also getting activated in proestrus. And what I have done here is that I have removed the ovary, so the estrogen will be lost. So you can see there is no light in there, and in males also there is no light in there. So you can make a comparison. This is a physiological expression, concentration which is elevated in proestrus. You can see a good light, but when it gets off, when there is no estradiol, there is no light. So this is a very versatile model which you can calculate. Next slide, please. And I've done a pharmacological validation also. So here what has been done is an animal is overectomized to ward off the endogenous estrogens. So then the animal is weaned, it is kept for some time to recover from the surgery, and then the estradiol pellet is being administered to the animal. So you can see here the light is very low, and suddenly when you start injecting the estradiol pellet, the light is increasing. So again, this is a very foolproof methodology where you can test your drugs which is closer to estradiol. So there are so many drugs that are being under experimentation by several companies, several top-notch companies, and very new next generation contraceptive pills have come up into the market. So this is one system where you can test all those drugs before to see if there is any functional relevance before even thinking about taking them to clinical trials. And you can also do an XY imaging. You can sacrifice the animal and you can take out all the organs, organ by organ. You can put it in a petri plate and you can make a readout of which organ that is getting activated to the maximum by the drug of interest. Say for example, here I administered estradiol and I can see a very huge light in the liver followed by reproductive organs. So something like that. Next slide please. So here is where your AI comes into play. So when you have a lot of data to compare, like we humans have only very minimal time to do that. When you combine this with the AI technology, the data with the AI technology, that AI can do this job and it can give you high throughput data. Say like your pharmaceutical company is giving you 100 drugs to test in a week, you can do the experiment and you can feed in the data and you can get the output as early as possible. That too meaningful output and statistics will be given by any AI. Next slide, please. So this model, here I am taking the example of a very mouse model. So this model can also be used to study endocrine disruptor chemicals and also to screen for functional effects. Next slide, please. So what the classical toxicity test? If you go to any pharmaceutical company, there will be a rat study, there will be a mouse study, there will be a rabbit study, there will be a legal dog study sponsored by pharmaceutical companies. So when it comes to traditional toxicity test, there is something called OECD guidelines, and there are several guidelines approved by the government and other consortiums. So you have to do those toxicity tests as per the guidelines. So there is something called classical toxicity test, which will report only over toxicity, but it will ignore the point of low doses. So whenever there is a low dose of drug or any endocrine disruptive chemicals or anything being given to the animal, what will happen? Like they tend to regulate the body. They'll try to overtake the situation, and there will be homeostatic regulation, and that thing will not be reported. That low dose toxicity will not always be reported because we are not dealing with a functional model in a classical toxicity test. So you cannot get the readout in a snapshot of time, and the next point is that there will be huge animal usage. Lot of animals will be employed in a classical toxicity test, and in fact you have to compare the species differences. Say like you are doing a test in rat, and then you have to make always a cross-species comparison, something like that. And another factor is also that some drugs may be active only in mouse, but will not be active in rat. So there are species differences also. So that is a problem with classical toxicity tests. But with this reporter mouse technology, it will be very functional. You can understand actually whether your drug of choice or drug of interest or drug in your hand is actually hitting your target or not. So this technology allows you to see observe the toxicity of any drug in a physiological setup. That is very important. Like you're not accrediting the system, at the same time you're not suppressing the system at all. That is the main important point. Like you will observe the activity of your drug of interest in a physiological setup with respect to time and space, as I showed you in the earlier slide, and you can actually understand the dynamics of toxicity. Say for example, what is the dynamics of toxicity? Say like your drug will not be toxic in one dose, but when you give the same drug again and again, because due to repeated bioaccumulation, it may become toxic. So such kind of an information you can easily get it from the reporter mouse. Next slide, please. So, this project I worked in with the European Union consortium, where they were very worried about the contaminant called cadmium that is present in the day-to-day food. I mean, in the Europe they take only bread as a staple food, like we take idli, dosas, etc., etc. But the main staple breakfast is bread. So they have actually they are very concerned about the amount of cadmium levels in the contaminated bread, and they wanted to test it. And this is being formulated as a feed to the animal that it is being given, and indeed we are able to see some we can able to see some low dose toxicities with the amount of bread that we have supplied. Next slide, please. So in that, we have not seen anything in the in vivo imaging, so that is what I showed in the earlier slide. But when you do an ex vivo imaging, you can see clear differences in the brain. So your cadmium contaminant is actually doing something to the brain area of the animal, which means that it will raise some questions like whether it is really safe to consume or not. Like, is there any way to minimize the level of cadmium? Is there any way to redefine the acceptable levels of cadmium? So, like this study will have given some inputs to the questions of redefining the standard of approach. Next slide please. So likewise, we have also tested immature male mouse. We have taken the reporter mouse and we have chosen the immature male mouse, and we have found that the contaminated bread with cadmium has some effect in the skeletal muscle, and in mature females they have some effect in white adipose tissue and thymus. So this is all about the power of this reporter mouse coupled imaging technology to rule out the subtle effects of dietary cadmium in bread. Next slide please. So now we all know, we are all taking soy milk because there is a advertisement in which John Abraham is taking soy milk. So there are so many brands which have come up in the market, and it is now taking green tea and soy milk has become a kind of fashion. But in our culture, there are so many good things which we have actually skipped now. So in the West, soy milk is very, very famous, and you can see that soy milk is behaving as equivalent as estrogen compound. So here you can see the lyophilized soy milk and soy mimics. So it is actually activating the estrogen receptor almost equivalent to a estradiol, endogenous estradiol or 17-beta estradiol. So this trend has been found both in an acute study as well as in a chronic study. Next slide please. So next thing is about DDT. So you all know DDT, we use so many pesticides at home to ward off insects in our kitchen. So what we did in our lab is that we compared this DDT with 17-beta estradiol, so which is a very standard hormone, positive control. So you can see here, although here it is 15,000 and it is 1,500, DDT is not as bad as estradiol, but it is it is not as good as well. It is also significantly increasing the liver estrogen receptor activity, which is not good for us. So this is reflected in the qualitative images also. So this DDT is doing something to the testis and also to the thymus, which means indirectly there will be chances of reproductive toxicity, and there will be some toxicity to the immunological cluster as well. So these are just simple experiments which you can do to know what is actually good for us, what is safe for us to use, something like that. Next slide please. So coming to the physiological applications of in vivo imaging, next slide please. So this is what I showed you earlier. A mouse has only 4 phases of menstrual cycle, and you can actually compare this menstrual cycle differences between 2 different models. Here I have made a comparison between AD and ID1 AD. So this is a sister model of the original model. So I can make a comparison on the various distress phases of the animal. Next slide please. And in fact, you can make a comparison between the age of the animal. So 1 month, there are 2 models here. So I am making a comparison between 1 month, 14 month and 21 month. And you can see here that the light is getting up. So with this technology, we can actually see the expression status of estrogen receptor with respect to age. Next slide please. And now I wanted to introduce another model called intermittent reporter mouse. So you all know that there is a cell proliferation gene called NF-Y, which is a master regulator of cellular proliferation. So what we have done here is that we have put up a luciferase gene in front of the cyclin beta promoter, and this NF-Y will strictly go and sit on the top of the cyclin beta promoter. So whenever there is a cellular proliferation, this NF-Y gene protein gets activated and it goes and binds to this promoter. So this is being thrown into the genome of the animal, and the animal was generated. And you can see here, this is a surrogate mother, this is a negative animal, but this is bearing because of positive embryos. So you just take out the embryos, and out of these many embryos, only 4 embryos are because positive. Means that they are transgenic positive, while the others are transgenic negative. And when the animal is in the embryo stage, you can see the light being emitted almost uniform in all the body compartments of the animal. But however, the heart is activated to the high level and femur is activated to the high level, which means the proliferation is very rampant in those organs. Go to the next slide. But you can see in the adult animal here, the cellular proliferation is very limited to the sternum and to the feet. So, and also a little bit in the limbs. So, why you see light in the limbs is because there is something called osteoblast which will be continuously renewing. So, which means that the cellular proliferation will be continuously happening because the animal will be crawling all the time. So, and here the sternum is growing because it is nothing but the bone marrow is being producing the blood cells. So, that's why you see cellular proliferation very much restricted to few body organs, unlike the embryos. So, now go to the next slide. So, this model can be used as a model of carcinogenesis. Next slide. So, here I have taken something called DMPA, dimethyl benzamethasone, which is a very traditional breast carcinogen. So, when I gauge the animal with dimethyl benzamethasone, like the liver estrogen receptor is getting activated within 6 hours, and the breast area is also getting activated in about 6 hours. Next slide, please. And you can see how the breast tumor is developing spontaneously. In baseline, there is no tumor. Slowly, the tumor is getting developed, and you can see it with your naked eye later on. And that, in fact, is being mirrored by the imaging technology. See, you can see capital here, and you can see the amount of light that is being emitted by this forming breast cancer tumors. So, you can even do an X-ray imaging to make a cross comparison of what you saw here is the real thing or not. Next slide, please. And you can do a dissection and you can see where exactly the light is coming from. And after DMPA treatment for 5 days and after so many days of monitoring the animal, we are able to see clear breast tumors in the mammary fat pad that in fact was confirmed by immunohistochemistry and histopathology examination. Next slide, please. And so in the earlier slide, I have administered DMPA internally by oral route. Here I have treated the animal on the surface. So you can see on the surface the light is glowing. I mean the carcinogenesis event is happening, and the photon emission is also increasing, which is being quantified. Next slide, please. So coming to the pharmacological applications of imaging. Next slide, please. So as I told you, we can study any drug that can bind to your target of interest. Here the example is, since we have an estrogen receptor reporter model here, we can actually study the estrogen mimics and see whether they have a potential activity on the endogenous estrogen receptors or not. Next slide, please. And here I made a CDX. CDX stands for cell line derived xenograft. So you have cell lines which you can get it from American type cancer cell collection, and you can inject it into the animal. Either you can do it orthotopically or heterotopically. So orthotopic now, say like you have a breast cancer cell line, you can put it inside the breast mammary fat pad, or if you want to study heterotopically, you can put it in the subcutaneous fat pad of the animal. So here I use a standard drug called tamoxifen. So tamoxifen is a standard drug for breast cancer. So once when you give tamoxifen, the light is coming down while the phosphate buffer saline, the control is starting to grow. And here you can see that tamoxifen treated animal tumor is going down, but this tumor is increasing again. Next slide, please. And here I started treatment with nicotinamide. So any pharmacy student here can tell me what is the real function of nicotinamide with respect to estrogens. Any pharmacy student can explain that. So the actual function of nicotinamide is with the other name for nicotinamide is niacinamide. So its function is to increase the estrogen status inside the living system. So here I got only negative results. There is no much difference between 0 hours and 6 hours. It is because 48 hours, 2 days. Because it takes even more time for the niacinamide to actually act to generate lot of estrogens within the animal. So that is the reason that you are not getting any signal, and that indeed is very practical and it is very real. So that is what I want to tell from this slide. Next slide, please. And in fact, you can also do brain sections, and like in brain there are a lot of regions. And if you want to do some neurological research, you can use this technology to do it here. For example, so I have compared the differences between estradiol treatment and vehicle, and this is IC is a potential antagonist against estrogen receptor. So here V2 is an agonist, IC is an antagonist, and here I have given a combination treatment. So you will see this light is getting totally zero here, but here the suppression has happened to the minimum. So something like that, like it's a very versatile technology that you can even use it for neurological brain research. Next slide, please. And now I want to introduce another model called PHARMACEUTICAL animal model. So here this stands for peroxisome proliferator activator. So when I put in a luciferase which can mirror peroxisome proliferation activation receptor, so there are so many companies which would come with some bench validated drugs. Say for example, here I used a drug called WY. So this has been given by a pharmaceutical company to test in this model, and you can see this PPAR has been activated in the liver, which means that so when you activate your PPAR receptors, your insulin secretion will be proper, and you your body will start to function normal, and you will not get diabetes. So in case if a person has a impaired metabolism due to diabetes, so this can help them. Like that's how drugs like pyruvate and all have come up in the market, which is basically anti-diabetic medication. So, but pharmaceutical companies are working so much in the field of diabetes, because year by year new recent generation drugs have been coming into the market. Next slide, please. So you can even do an ex-vivo imaging, as I told you before. When you give the PPAR agonist, it is getting the liver is getting activated. Say for example here. So you can see very clearly, and the data is very straightforward. Just by looking at the data, you can understand whether your drug has an effect or not, whether it is useful or useless. Next slide, please. And this I will skip because this is repeated here. And here I checked a drug called 3-beta-diol. So to check the activity of 3-beta-diol, so I injected the prostate cancer cell line, bioluminescent prostate cancer cell line. It is a prostate gland of male nude mice. So nude mice is something like it doesn't have a proper functioning thymus gland. So when the properly functioning thymus gland is missing, the animal will not be able to produce enough T cells to have an immune response. So that animal is a basic immune compromised animal. So when you put a cell line, cancer cell line into that, the animal will start to grow those tumors. So this is what we are seeing here, and this is a placebo treated animal, and you can also see some metastasis here and there. But when you give 3-beta-diol, and you can see the light is totally going down, and the chance of metastasis was also being reduced. Next slide, please. And this, I treated the animal model, the liver with 5-fluorouracil. So 5-fluorouracil is an anti-cellular proliferation agent, and it's an anti-cancer drug principally. So when we administer 5-fluorouracil, even the light in the sternum, the cellular proliferation that is happening in the sternum is going down with increasing in dose. And we actually is nothing here. And why do you see a spleen here? Anybody can answer this question. Why are you seeing a spleen that is growing here? It is because cells, blood cells, spleen is the graveyard of the blood cells. So you can see the blood cells that they are in the phase of dying, and they are getting populated into the spleen of the animal. Next slide, please. So likewise, like here I have treated with the mucilide. So this is a wound healing drug. So when I hit the cutaneous wounds with mucilide, I can see the activation of the cellular proliferation. The wound is getting healed. So that is all about the pharmacological applications of this reporter model with the example of mucilide. Next slide, please. Next slide. So this model can also be used as a model to study the cellular, molecular and developmental biology fields as well. Go to the next slide. So you can see the wild and pregnant mice with this is cellular proliferation with the state, and you can see the light increasing, and slowly when the animal is in an advanced stage of pregnancy, you can see the pups which is becoming positive, transgenic animals which is growing under a transgenic negative mother. Next slide please. So you can actually study the model in each and every life cycle of the animal model. So starting from day 1, you can study up to 99.5 DPC or even more than. Next slide please. So coming to the conclusion, this in vivo imaging technology is a very, very versatile technology. It can be applied to any basic and translational research field. Especially, this imaging technology is a state-of-the-art technology which will be very, very useful in the field of pharmacology. And with this technology in hand, they can actually reduce the attrition rates. So attrition rates, so many drugs are being studied by pharmaceutical companies. So the chances of success rate of the drugs will be a success rate of this technology will be very, very useful, and you don't have to invest more money. Say like for a drug activity, then why you go for the next next phase of the testing the drug. So with this itself, you can get enough information of your drug of question. So it's such a versatile technology, and it can be applied to any field as well. And this technology is expected to accelerate the way of the drug discovery and drug development programs are being conducted. So next slide, please. So I have talked enough about in vivo imaging technology. So one image is what about talking a lot actually. According to me, next slide, please. So next slide. According to me, this is the image that I will say is 1,000 words. And thank you so much for listening to me with patience, because it's almost 1 hour that I was talking to you all. And thank you so much, and I'll be happy to answer your questions if any. And you can even write to me if you have any idea of doing PhD. But provided that you should have a self-supported fellowship. We don't take students without any self-supportive fellowship, like you can write a CSAR or CSAR NET or ICMR or DPT JRF, or there are so many other courses. Like women, they can apply for Vice Kiran Fellowship, and then there is another thing called Welcome Trust. There are so many competitive exams that you can clear to get your own funding source for your salary. So if you guys have cleared any such examinations, you can always approach me to have a PhD thing being done under in our department or under me or anything. Thank you so much. Thank you so much.
On behalf of everyone present, I would like to extend our greatest gratitude to our Balaji Ramachandran sir for such an enlightening and thought-provoking session. Your insights into revolutionizing the drug discovery through AI undoubtedly broadened our understanding, and we sincerely appreciate your contribution to today's conference, sir. At this time, we will open the floor for a brief Q&A session. We invite you to share your questions with our speaker. Please raise your hand and our team will assist you with the microphone. We ask you that questions remain focused and concise to allow meaningful engagement. Let us proceed with Q&A.
I think it's crystal clear to everyone, it seems, or like they may be hungry. But whatever I have presented here, it will be very useful for people who wish to pursue a career in the field of pharmacology especially. Don't you guys have pharmacology practicals? So how do you do those practicals? You do the software simulator.
ஏதாவது கொஸ்டின் இருந்தால் கேளுங்க.
Sir, how is this useful in any disease diagnosis, sir?
See, this is not a diagnostic methodology, first of all. You cannot diagnose a disease, but you can use it for preclinical research. You cannot use a model to diagnose a disease, first of all.
Thank you for the response, sir.
Okay, then, if there are any more questions, I think I can sign off. Thank you.
So on behalf of everyone on the dais and off the dais, I would like to take this opportunity to thank Dr. Balaji Ramachandran for his enlightening session. Thank you so much, sir.
అల్లే ఉన్నా
Welcome back everyone. I hope everyone enjoyed a refreshing meal. As they say, a filled stomach fuels a focused mind, and we are excited to continue with the next session of the day. Let's reconvene with renewed energy and enthusiasm as we move forward with the next session.
Leadership is not about a title or a designation. It's about impact, influence, and inspiration. Leadership is about getting results, spreading passion, and inspiring others. We feel more powerful with this leadership. We are privileged to have an enlightening keynote speech from our esteemed chairman of Sree Sastha Group of Institutions, Professor J. Karthikeyan sir.
Institute of Science and Technology, Chennai, beloved principal, Dr. Dhanapal, faculty Professor Dr. Mubarak, Professor Shamsundar, faculty members, and my dear students. Good afternoon to one and all here. I should have been a speaker of yesterday. Due to my sudden some developments, I was not able to participate. Today morning also I told him, today also morning I had another meeting outside. So I disturbed the whole program now coming into as a speaker. Yeah, I should appreciate all the primary idea came from Dr. Dhanapal. Wanted to conduct an event, and he discussed with me. Later on, all the faculty members, coordinators, event conveners, coordinators, everybody met me and they explained, and they have demanded a budget from the management. Apart from the collection from the students, we readily agreed. Sir, as yesterday Dr. Babu was mentioning about why you need all these sort of conferences and seminars and celebrations like Pharmacy Day, other days. You all know, it is again what he said, I am recalling, it is only bullet. You need a gun. Gun is this conference. Apart from academics, whatever you have to learn, these are all opportunities. You want to hear Dr. Abdullah on his special day speech, or want to know what are his presentations, you can get into the tube, you can learn it. You can hear them, and you can see what is the good about it. Similarly, you can see all of them. So these sort of things, it is an opportunity, no doubt. It is all available in the net. When you search for it, you find it. Nothing like gathering and hearing in person. A music system or a music concert where you go, you listen to the music, is entirely different. That environment makes you to enjoy more and you get relaxed. Stress-free, known friends, unknown friends, you meet there and you discuss. Here, this is a technical meet, a conference where you will have a lot of inputs from the students from outside college, what they are doing there, what we are doing here, what is the good thing that we can take it from them, or from that college, or from the principal. I think many faculties also have enrolled here. You can hear them, talk to them, what is good about it, what is the industry is doing. These are opportunities only in a conference you can get it. So try to make use of this 3 days conference, be attentive, try to attend to the speaker's note. I don't find anybody carries a note. Students, please bring a notebook with you, whatever it is that you find here, or by talking, by hearing to the speaker, you would be interested in visiting a site. You can take down, they refer so many sites here. Take a note of it and try to go to your place, home, hostelers or day scholars, try to take a note of it and just have a look at it. Maybe you are in 3rd year, 2nd year, 1st year. You can just look into it. What is that sir has told or the speakers have told. Get into that net, their website, they look at it or the area. Maybe Pfizer will be working on that area. very seriously. If you source it in the net, you can find that Pfizer is working. If that is your area of interest, follow him. Like in LinkedIn or in Facebook, follow him. See continuous posting of that company and what is the development they are reaching. And it will give a fairly good idea. I want everybody to become a Facebook and LinkedIn members. I want, last time we initiated it, I think most of them are there. And what you follow is very, very important. Don't follow Nayanthara and Rajini. That is okay, that's for 5 minutes. You follow your professional people. Professors are coming on the line. Somebody from US and somebody from Switzerland is coming on the line. They post their findings and research details. At least a minimum. Today you have ChatGPT. You can find out anything, any meaning, any moment you can take it. See, these are all wonderful opportunities. I wanted to tell you, really at this moment, I used to ask myself, I should have born in as a millionaire, not as an 60s, 70s child. Because now you have plenty of opportunities, new things have to learn. Every day, my family also used to ask me, why when you get up, you start looking at your LinkedIn postings and other things. It is not my posting I am looking at it. I am looking at all the professors and the companies, what they are doing, how India is behaving, how the growth is going to happen. Definitely India is growing, nobody can say no. Apart from small issues here and there, they have been placed very well today. In today's issue also, morning when I was seeing that, it is they are negotiating with Russia and Ukraine to solve the problem, war problem. Same way they are trying to talk with, trying to be a mediator for this Palestinians and Israelis, that is with PM Netanyahu. So many things are going, these are all taking us in the better position of the country, which means they create confidence within the third world nations. They look India as a very prospective, bright opportunities for their companies also. And they don't have, they want to produce, but they don't have, what do you call it? They don't have this sort of manpowers, this technical, qualified. This age group we don't have in anywhere in the world, apart from China and Vietnam. Vietnam, okay, they are lower than them, but Chinese, as student, as a business entrepreneur, as a professor, we have to learn a lot of things from China. China is not only for the material, the technology. Somebody would have read today what they are talking about. In Biden's government in the US, they wanted to remove all Chinese cars. Here also the Chinese cars are coming, BYD. Those cars they wanted to remove it. Okay, China said, okay, we'll remove it. Now next things, they are enforcing, though we don't want to buy any parts from China. Because all Chinese parts are it's a communicatable and the Chinese people can know what you are doing. So this should not happen. This is the idea of US. So they said we will not buy any parts. They have a target. From this year onwards, we will not buy any parts from China, any electronic parts. So this sort of a threat is there. So that's confidence. But they need the material. Who is going to give it? In the world, next manufacturer is India. So definitely they have to look back India, whether to buy the products or to establish a factory here and see that for the next 10, 20 years they don't have any problems. And they want to put China down. US wants to put China down. And definitely they are working on that. And the next government also, you are all aware, Kamala Harris and Trump is there. Trump is also against China. Then he is very much worried about this coronavirus. There is a myth that is being spread by Chinese people, and that is what he is totally dead against it. Madam Kamala Harris, okay, she has a concern about it, she has to talk more about it. Why I am telling all these third world countries is, everybody is looking at India. So we have a very beautiful opportunities. Students, make use of it, spend time on social media. If you want to grow in that area, follow that company. If you want to grow in that area, follow that company. Definitely you will have larger reviews and you will gain more knowledge. And the salary was around 5,000. Imagine the salary today and that point of time. So what is today? Everything is available. Skill is appreciated. Everything is available. Skill is appreciated. skill is being brought to the light, skill is recognized everywhere, and you will get a better payment. So these sort of conferences are definitely very good, and mostly in Corona period is all on online. Now we have both online and offline. Webinars are being conducted. You call a US pharmacist and you want to talk, you can talk. You can display it in your web classrooms. You can listen to him. What is he telling? What is that in the LinkedIn telling? What is a teacher telling? What a research lab is telling? You can just come. This morning I was there in a meeting. I was just looking at something. In Google there is a place called Search Lab. Search Lab. You can easily find out. You ask for any details, it is giving on the laboratory. So the world is totally open for everybody. Now you have to take your positions, where you are going to stand and how you are going to perform. With these few words, I congratulate all the department, the College of Pharmacy faculty members who took a 3 days train is not an easy task. It is 3 days maintaining the crowd and handling them and advising them on day-to-day events. It's not a simple job. Wonderful, hats off to you, and you should congratulate the department for whatever you have done. So, take use of the opportunity. Thank you very much.
Thank you, sir. Now we call upon our beloved chairman, sir, to felicitate the chief guest on behalf of our institution. Sir.
ఫ్లాన్స్ వెళ్ళరా
I would like to call upon someone who always brings warmth and enthusiasm to the stage. As the saying goes, a cheerful heart is good medicine, and no one embodies that better than Dr. Saranya. Please join us in welcoming our esteemed speaker, Dr. U. Ubaidullah, for the evening session. Over to you, ma'am.
Good afternoon to one and all present here. I am here to welcome the esteemed speaker of our today's afternoon session, Dr. Ubaidullah. He is an associate professor in the Department of Pharmaceutics, Crescent School of Pharmacy, B.S. Abdur Rahman Crescent Institute of Science and Technology, Chennai. He has more than 21 years of experience in the profession of pharmacy. He has contributed extensively to the world pharmaceutical technology literature with a score of H-index 90 and Scopus index 60, with publications appearing in reputed journals. He serves on the editorial boards of reputable journals and the doctorate committees of several prestigious universities. He worked for 5 years as a manager in Dr. Reddy's and Lupin Pharma. He has 3 international patents and 2 Indian patents to his credit as principal investigator. He is guiding many PhD scholars in the field of pharmaceutical sciences. He has guided more than 40 M.Pharm and many B.Pharm students for the project as a part of their curriculum. He has published 5 textbooks and more than 70 research papers, as well as been awarded funding projects from national and international universities. It is a pleasure to welcome Dr. Ubaidullah to deliver today's keynote on AI's prominent directives towards designing DDS and PT. We are honored to have you, sir, in sharing your insight with us. Thank you, sir.
Thank you, ma'am.
Light off. Light off.
Now I request our speaker to move forward for his presentation. The stage is yours, sir.
Respected Chairman, sir. Respected Principal. Good afternoon, Nana. This is here. respected chairman sir and principal sir, our colleagues, faculty colleagues and students who are present here, very good afternoon to everyone. All of you happy now? Feel free now? So this is one of the important topics. Just to your chairman sir told, only the few points, the business. So you want to empower the community or India, then you have to be in the business. All the third world country will be looking for the India. India have the very skilled people and when compared to other country, we have a lot of skilled people, but the skills should be advanced one. So your principal sir choosing the title, Accelerating Pharmaceutical Innovation. Pharmaceutical innovation is a old concept. Now we have to be accelerate or progressive innovations. So what do you mean accelerating? Accelerating nothing but, so you are doing the innovation innovation in the pharmaceutical industry for preparing tablet, capsules or whatever dosage forms. It takes time. It takes 6 months time to develop the one dosage form. But what is accelerating means? You have to perform the same product with a short period of time. Is it possible? Yeah. Is it possible to accelerate the pharmaceutical innovations, students? Is it possible? How many of you say yes? How many of you say no? Accelerating is possible or not? Yes, sir? Very good. So the accelerating word is very suitable for artificial intelligence. So the AI will be helpful to accelerate all the process, either pharmaceuticals or engineering or agriculture. So every field, the AI will be helpful to accelerate the innovations. So you all remember that. So listen here. This is one of the famous magazine published this picture. So the magazine, what it says that the pharma company should collaborate with all other AI companies and tech corporations. Then only we can go for accelerating the innovation. If you are alone for doing the innovation, we cannot grow much. So listen here, the Pfizer, AstraZeneca, Merck, Novartis, all of the company, all the big player of the pharmaceutical company has to collaborate with AI companies. AI companies, Cyclica, E-Therapeutics, Cloud, Unitas, etc. Very big AI companies. The tech corporations, you already know, IBM, Intel, and all. So what the magazine says that the pharmaceutical company, if you want to really grow, if you want to compete the world, the companies should collaborate with AI company and tech company. Then only it is called accelerating the pharmaceutical innovation. If you simply keep on working for developing the tablet in a world company, it is not accelerate. If you want to really accelerate, then you have to be open to work with all, collaborate with all. Collaboration is very important. So here you can see the sentence of Forbes. So do you know what is Forbes? Forbes magazine. It's one of the famous magazine in the world. You can simply label this. So the data, what they publish is, you can label one. So what it says, the impacts of the drug molecule with the biology system has required the pharmaceutical industry to develop sophisticated flow where any human expert make your decision based on the experimental data. So here, எல்லா கம்பெனியும் மற்ற கம்பெனி எல்லாம் வந்து டெக் கம்பெனி மொபைல் கம்பெனி எல்லாம் வந்து they won't interact with the human population. Only for the pharma company only has to interact the cell systems. So you have to interact once you interact the bio system, you will get a lot of other side effects, even in the toxicity, so a lot of other effects also is possible. So you have to take care of the population so that you have to create a lot of experimental data. The data, once the AI proves that the ability to solve the complex problem, then the whole industry can be transformed in totally different business. So in the problem which is solved by using the AI, you will get a lot of other business. So that you will transform the complete business of pharmaceutical industry. Listen, are you understood? Clear? Read out. Students, it should be interactive session. It should not be lecture. Okay, I don't know lectures. I should be you go for interactive sessions. So you can interact. You can also talk. You can also participate in the call. So what you are doing is any students can tell the answer? Anyone tell the answer? What is artificial intelligence? What is artificial intelligence? Students, anyone? Anyone? Give the mic. Give the mic to anyone. Otherwise they keep on moving passive. So we have to keep on intact with the students. Because it is this artificial intelligence world, maybe first year or second year, third year, fourth year, your syllabus is not there. So you will get bored, you have to understand anything. So if you keep on talking now, you will get bored. So that's why I keep on impacting you. So whatever the doubt you have, you can just ask during the session. So the simulation of the human intelligence process. What is simulation? Simulation is nothing but what are the process you are thinking in the brain. That concept should be available in machines. The same concept you can replicate in the machines without exposing the human populations. So all the pharmaceutical industry, what are the drug molecule, what are the things you are using, everything it has to be intact with our biology system. But we cannot expose the biology system simply without proving the concept. So for proving the concept, you have to always develop the simulation technique. Simulation technique is nothing but without using the human population, what your brain thinking is, you have to bring it to the machines, like computer system or whatever it is. So how it is possible? The process is first the machines should learn it. What you are thinking in the brain, the machine also should think the same concept, then reasoning. So what your brain, you can say the brain thinking is something like that. If you develop this product will be helpful for anti-cancer. So the same type of thinking should be the machine also thing. The last one is the self-correction. That is very, very important. So the machines, they find any fault in the process, the machines have the capability to self-correction the process or results. So the three processes are very, very important one. The first one is the learning. The machine should be learned, and it should be reasoning and self-correction. The movie when you ready pannina, that is called artificial intelligence. Simple சொல்லணும்னா அந்த மிஷின் வந்து என்ன பண்ணும் எப்படி பிரைன் திங்க் பண்ணுதோ அதே மாதிரி வந்து என்ன பண்ணும் லேர்ன் பண்ணனும் அதே மாதிரி என்ன பண்ணும் திங்க் பண்ணனும் ரீசனிங் கண்டுபிடிக்கணும் அதே மாதிரி என்ன பண்ணனும் செல்ஃப் கரெக்ஷன் பண்ணிக்கணும் அது தன்னை வந்து என்ன பண்ணனும் திருத்திக்கணும் இது மூணையும் பண்ணுச்சுனா that machine is called as artificial intelligence. சிம்பிளா சொல்லணும் புரியுதா? ஃபர்ஸ்ட் வந்து மொத்தம் நாலு வேர்ட் தான். ஃபர்ஸ்ட் வேர்ட் இஸ் சிமுலேஷன், third second word learning, and self-correction, last self-correction, reasoning, self-correction. The terminology vachina, the whole concept of artificial AI is working. Clear? Now, and explainable AI, there are two terminologies, the different terminology. AI is the machine. One can understand the process and completely it will give the results. But we need the explainable AI. Why? Because the AI only giving the results and you cannot, the machines cannot cut the error. Understand why? And learn and correct the errors and read and down demand. So based upon demand of the merit, it should be changed. That is called explainable. We want, mobile is, if someone is calling you, the mobile giving the name of the call. If it is not giving, then what is the use of the mobile? You have to find out who is calling. So like that, the mission should be understand the concept and it should be giving the results and based upon the demand, based upon our purpose, it has to be reenact. The moon panada, that is beautiful AA, that is called actionable AA. புரியுதாப்பா விஷயத்துக்கு வந்து என்ன பண்ணோம் ஒரு ரிசல்ட் கொடுக்கும் வேற ரிசல்ட் தப்பா கரெக்டாங்கிறது என்ன பண்ணோம் as a human expert தான் we have to understand இப்போ இருக்க டெக்னாலஜி எல்லாமே மொபைல் இருக்குன்னா மொபைல் வந்து கரெக்டா தப்பா என்ன பண்ணோம் as a parent we have to be check it and we have to give to the kids child we have to check each and every day we have to check the mobile for the mobile while using the kids that is our duty but the AI should be the if suppose giving the kids that what are the websites lock it or what are the things the students should see it everything should be controllable by AI so you need such type of AI to control it and monitor it and reenact it that is called actionable AI.
Hello? Oh, yes.
How many have to understand AI? கை கொடுத்தப்பா யாருக்குள்ளப்பா ஏன்னு என்ன புரியுது மொத்தம் 600 பேர் இருக்கீங்க இல்லையா how many have to understand AI how many have to not understand still AI புரியுதுப்பா தமிழ்ல தான் பேசுறேன் I can also deliver to Tamil also if you want ஏதாவது சொல்லுங்க சொல்லுங்க AI னா தெரியுதா so AI nothing but ஒரு சிமுலேஷன் ப்ராசஸ் ஒரு மெஷின் தான் அந்த மெஷின் வந்து நம்மளுடைய பிரெயினை வந்து மேப் பண்ணும் எப்படி மேப் பண்ணோம் நம்ம பிரெயின்ல என்ன திங்க் பண்றோமோ அந்த கான்செப்ட் வந்து என்ன பண்ணோம் இந்த மெஷின் வந்து ப்ரொடியூஸ் பண்ணும் அது கான்செப்ட் வந்து டேட்டா ரெடி பண்ணி அந்த டேட்டா வந்து ரிசல்ட் ரெடி பண்ணிட்டு அதுக்கப்புறமே அந்த ரிசல்ட் வந்து ரைட்டா தப்பா நம்ம கொடுத்தோம்னா இட் வில் சேஞ்ச் த டேட்டா இட் வில் சேஞ்ச் த ரிசல்ட்ஸ் தட் இஸ் கால்ட் எக்ஸ்டர்னல் ஏஐ புரியுதா நெக்ஸ்ட் so why the AI is very important in pharmaceutical industry எதுக்கு எல்லா இடத்துலயும் ஆர்டிஃபிஷியல் இன்டெலிஜென்ஸ் பத்தி கான்செப்ட் நடத்துறாங்க வொர்க்ஷாப் பண்றாங்க நிறைய எங்க பார்த்தா AI சொல்றாங்களே what do you mean why we need AI because next generations next you can say next for 10 years all the industry have the AI because they have to accelerate the production accelerate the innovation accelerate all the process so it is a progressive world so you have to be compare the world compared to China we should also be compare the fasting the process by developing the product so in pharmaceutical industry these are the area where AI can play the major role ஒரே ஒரு ஏரியா மட்டும் இல்லப்பா எல்லா ஏரியாலயுமே வந்து AI வந்து play major role first one is clinical trial design and monitoring clinical trial what is clinical trial கேள்விப்பட்டீங்களா what is clinical trials ஒரு ட்ரக் ப்ராடக்ட் வந்து ஹியூமன்ல கொடுத்த பிறகு அது ஹியூமன்ல வந்து அந்த எஃபிகசி ப்ரொடியூஸ் பண்ணுதா இல்லையா இப்போ டேப்லெட் எடுத்தீங்கன்னா அந்த டேப்லெட் வந்து என்னது சப்போஸ் வந்து டேப்லெட் எடுத்தீங்கன்னா it should reduce the blood sugar level so அப்ப என்ன பண்ணோம் அந்த டேப்லெட் வந்து blood sugar level குறைக்குதா பார்க்கணும் so அத பார்க்கும்போது நம்ம எத்தனை பேஷண்ட் எடுக்கிறோம் எவ்வளவு டேட்டா கிடைக்குது அதனால it will take a year process it is not a single process ஒரு கிளினிக் என்ன பண்ணணும் phase 1 now take 2 years 3 years time so if you want to use the time if you want to accelerate the process then you have to use the AI to collect the data of a faster pace. The next one is the QA and QC. What is QA? For the students, what is QC? Next one QC is the quality control. So for QA and QC, if you want to process the preparation of tablet, the QA QC has to be approved. The process, what is the process you follow, the QA QC reports should be approved. So what the role? They have to analyze the sample and they have to release the specification. Then only the product will be reached to the market. It takes another 1 month time. If you want to reduce the time, then you have to use the artificial intelligence to foster the analytical process. The third one is pharmaceutical manufacturing. So manufacturing now you know tablet, compressor, tablet, capsules, syrup, it will be used for the various type of equipments. The equipments on the process on the fast part of the way, we can use the automated one. So that it will be controlled by the machine, computer, from that we can easily check the process. The fourth one is pharmaceutical product management. What is product management? What is product management? So the unique property of pharmaceutical product is every 2 years or every 2 years the product will be expired one. So once the product will expire, there is no the market demand will be increased. So you have to analyze the expiry date and availability of the product in the market. Based on that, you have to design the production process. புரியலையா? நிறைய ப்ராடக்ட் நீ manufacture பண்ணி single day dump பண்ணி என்ன ஆயிரும்? எல்லா ப்ராடக்ட்டுமே அந்த சேம் டைம் எக்ஸ்பைரி அப்ப மார்க்கெட்ல வந்து போட்டே இருக்கு. சோ ஒவ்வொரு ப்ராடக்ட்டையும் என்ன பண்ணணும்? கரெக்ட்டா டைம் லைன் ஃபிக்ஸ் பண்ணி you have to fix the time line based upon the expiry date you have to manufacture the product. Then only the product available in the market at a regularly. If not available regularly, what will happen? ஒரு ப்ராடக்ட் இருக்கு, அந்த ப்ராடக்ட் வந்து மார்க்கெட்ல வந்து இருக்கு, கொஞ்ச நாளைக்கு இருக்கு, கொஞ்ச நாளைக்கு இல்ல. அப்புறமே கொஞ்சம் இன்னும் so the patients change the brand. So you lose the market. The company lose the business, lose the income. எல்லாமே என்னது? It will be disadvantage of the company. So the production management is a very important one. So the AI can be accurately determine the date and it will use the proper date to develop the products. Next one is the pharmaceutical product development. What is product development? So every year there are new molecules will come to the market. That molecule can be converted into tablet, capsule, let us solution, syrup, without development. So each and every company develop the product, but the company has to check it, the product is stable or not. Example, some new molecule, the company develop the suspension product. So what is the problem in suspension? It forms the hard cake. Now suspension is not particle on the settle hard cake form. So it forms the hard cake. So if you want to avoid this problem, then you have to analyze the product very fast. So you have to analyze the product. You have to ensure that the product is stable in the market. So for designing the product development, the AI plays the major role to assess the faster stability period and faster quality control testing. The last one is very important one, drug discovery. What is discovery? What is discovery? Finding the molecule from the paper to lab. Okay, first the molecule வந்து first paper design பண்ணுவாங்க. Then it will be bring to that concept bring to the lab. Then the people will be optimize the compound. There are number of compound will be done. Will be prepared in the lab. Then only one molecule will go for the clinical trials. So for drug discovery, it takes one molecule to reach the market almost 25 years. So drug discovery வந்து 20 வருஷம் ஒரு மாலிகுல் வந்து கம்பெனில ஒரு மாலிகுல் டெவலப் பண்ணி அதை வந்து ரெகுலேட்டரிக்கு அப்ரூவ் பண்ணி மார்க்கெட் வந்து how long take 20 years. So for the discovery, for leading the optimized molecule, we can use the AI to reduce the number of errors. So you can, the lead optimizations will be done by the simulation software, so that you can easily optimize the single molecule very fast. So all the area, the AI will be more helpful. Next. So the previous slides we used to talk about the generally how the AI is used. Now we are deeply discuss about in the pharmaceutical manufacturing only, in industry only, because the industry is the main primary objective of very primary number jobs creation post of the pharmacy. So where the pharmaceutical manufacturing we are using the AI. புரியலையப்பா நம்மளோட என்னது மேக்ஸிமம் பீப்புள் will go for the company தான். கம்பெனிக்கு போகும்போது நம்ம என்ன இண்டஸ்ட்ரியில வந்து எங்கெங்கெல்லாம் ஏஐ யூஸ் பண்ணலாம் அதை தெரிஞ்சுக்கணும். உங்களுக்கு இன்டர்வியூ பண்ணாலும் யூஸ் ஆகும். ஸோ இந்த கம்பெனியில் வந்து ஃபர்ஸ்ட் ப்ராசஸ் என்னது? ப்ராசஸ் டிசைன் அண்ட் ஸ்கேல் அப். என்ன ஸ்கேல் அப் என்ன? வாட் இஸ் ஸ்கேல் அப்? வாட் இஸ் ஸ்கேல் அப்? ஆர்டி லெவலில் பண்ணுறது வந்து அப்படியே மேனுஃபேக்சரிங் சைஸ்க்கு மாத்திரம் தான் ஸ்கேல் அப். ஆர்டியில் வந்து என்ன பண்ணுவீங்க? ஒரு ஸ்மால் பேட்ச் சைஸாக ப்ரூவ் பண்ணுவோம். அதை வந்து என்ன பண்ணுறோம்? கன்வெர்ட் டு லார்ஜ் சைஸ் ஆஸ் பர் த மார்க்கெட் ரிக்வயர்மெண்ட். மார்க்கெட் ரிக்வயர்மெண்ட் அந்த மாதிரி என்ன பண்ணுறோம்? We are preparing the tablet. Scale up nothing but R&D formula converted to manufacturing formula. Simple சொல்லணும்னா R&D formula converted to manufacturing formula. So here the process design. What is process design? If you want to develop a tablet, okay, the tablet you are using the different type of process. Either you go for the wet granulation or dry granulation, or you can use starch or PEP as a binder, or you can use another magnesium or tall. So there are a lot of factors playing a major role. So you have to choose the which are the inactive ingredients can be useful for the particular product. So this and all you can easily optimize by the AI to get the quick results. Which are the inactive ingredients mostly suitable for the particular tablet, and whatever process, wet granulations or another dry granulation, diet compressions. So இதெல்லாம் என்ன பண்ணலாம்? You can optimize by using the AI technology. Next one is the advanced process control. So sometimes the product will be having any if you want combination of the product, or sometimes you are using some advanced technique, then you have to use the AI to simplify the process. Suppose the AP is the poor water solubility. Poor water solubility, but the drug is insoluble water. Now you want to develop the tablet. Now you got to improve the solubility of the drug. So improve the solubility drug. What will you do? Anyone can tell how to improve the solubility drug? How to improve the solubility of drug? Size reduction, or you can reduce the pore solvency, or you can reduce the surfactant. So if the process is there, so you have the three ideas. Size reduction போலாமா, இல்ல pore solvency மெத்தட் போலாமா, இல்ல surfactant ஆட் பண்ணலாம். So இந்த மூணுல வந்து எது பெஸ்ட் எப்படி கண்டுபிடிப்பீங்க? How to find out which is the best process? You have to perform the process in the lab. It will take some time and waste of the time, waste of the product. But by using the AI, you can identify which process will give the best result. So next is the process monitoring and the fault detection. So this is a very important one. Listen, the fault detection. While during the manufacturing process, suppose if do you know what compression machine? Have you heard the term compression machine? பாத்தீங்களா? Tablet compression பண்ணுறோம் இல்லையா? So tablet compression பண்ணும் பொழுது the tablet weight variation may be changed, keep on changing. If suppose the weight variations go beyond the limit, then you have to find out, you have to switch on the instrument. The manually every time you have to collect the tablet and you have to get the weight tablet and you have to find out whether the tablet will be, the weight of the tablet will be within the limit or not, you have to check it. This is a manual process. This process only recently we are using now. Okay, boy, our offer. So the fault detection, if there is a compulsory missing connected with the AA process, the AA will inform you there is a weight variation which is there, it will be directly switched on the instrument. So you are completely avoiding the error in the tablet process. Next one is the turn monitoring. So based upon the turns, you can develop your AI and you can find out the solutions. Next, data integrity and data quality, the Internet of Things and data management, regulatory and process control, release testing, and the real-time data. So these are all the very, very important process. We can easily, the product management can be possible in the manufacturing industry. Next. So listen, please state for the reason. So the AI will be helpful for the pharmacist. One is the design and formulation and test and analyze. And please, first you have to design the product. Whether the tablet can be tablet or capsule, or the suspension tablet or the dairy tablet. So whatever tablet you want to produce, first you have to design the formula. Then you have to formulate the dosage form. Then you have to go for testing the dosage form. Then finally go with the analysis process. So all the 4 areas, the AI plays the major role. Next. So this is for only one example. So how the AI will be kind of helpful for the production. You can see what if ANN calculating ANN for the ANN, artificial neural network. So this is one type of the AI. There are different type of AI is there. So ANN is the proponent. ANN is the one type of AI process, artificial neural network. So the ANN collects a lot of data. The data will be predicted and go for the controlling process. Then you go for the formulation process. Then the formulation process, there you can check it whether the formulation is correct or not. If the formulation process want to adjust any parameter adjustment, here the AI can be advised you to change the parameter. Then if suppose the whatever control will be there, it will be good for you, then directly you can go for the formulation process. First the AI will be all the data, it will do the results. Based upon the results, you can go for the adjust the parameter, or if it is good results, then directly you can go for the formulation process. So what you do for the designing the process. So ANN will help you to design the complete process, so it will give the final product very easy. It is a short period of time. So ANN will help you to bring the final formulation within short period of time. The artificial neural network will helpful to final form to optimize the formulation with a short period of time, with a lesser time. So ANN and the final formulation will give the best results. Okay, next. So industry what do you mean cheaper production? எவ்வளவு கம்மியா காஸ்ட்ல வந்து இந்த டேப்லெட் பண்ண முடியும். இதான் வந்து ஃபர்ஸ்ட் டாக்கர். ஒரு இண்டஸ்ட்ரி ரன் பண்றாங்கன்னா இண்டஸ்ட்ரியல் மேனேஜ்மென்ட் என்ன பேசுவாங்க? ஒரு ப்ராடக்ட் டெவலப் பண்ணும்போது எவ்வளவு சீப்பா அந்த ப்ராடக்ட் வந்து டெவலப் பண்ண முடியும். சோ அப்ப நம்ம சீப்பர் ப்ரொடக்ஷன் வந்து எப்படி நம்ம ஏஐ வந்து யூஸ் ஆகும். இதெல்லாம் வந்து ஃபியூச்சர்ல வரும். ஏன்னா ஃபியூச்சர்ல வந்து the future is going to be AI playing the major role to reduce the cost of the production. So how the AI will be reduce the cost of production? See the diagram. So the A will be giving the proper results, whether what type of equipment you are using, it is correct or not. You collect a lot of data. From the data, it will give the results. So what type of instrument you have to use and what type of process you have to follow. Based upon that, you will get the correct product and within the correct period of time. So you can reduce the it will be reduce the vast process and it will give the simple process so that you can reduce the cost of the product. So every the A is the A will give the simple process. Now we are going for the tail method, red granulation, granulation, and the process the A will be sorted out and reduce the cost of the productions. So this is one of the very important A and a popular AN. AN is one of the, when compared to other AN is one of the popular one. And in pharmaceutical industry, most of the industry people are using AN, artificial neural network. So artificial neural network model involves computation mathematics which simulate the human brain process. So when compared to other process, the AN having the perfect matching of our human brain process. It is a multi-layer. When compared to other process, the AN is the multi-layer process where you can match our human brain process. See the diagram now note putting this is the basic concept of AA. So what are the input you are giving x1, x2, x3? So என்ன திங்க் பண்றீங்களோ அந்த இன்புட்டை வந்து கொடுக்குறீங்க. ஒரு டேப்லெட் பண்றீங்கன்னா டேப்லெட் வந்து என்ன தேவைப்படும்? லாக்டோஸ், ஸ்டார்ச், மெக்னீசியம். இது மூணு தேவைப்படுது. இந்த மூணு வந்து எவ்வளவு குவாண்டிட்டி தேவைப்படுது அப்படிங்கிறது வந்து நீங்க வந்து கொடுக்குறீங்க. அந்த மூணு இன்புட் கொடுத்தீங்கன்னா அந்த A என்ன பண்ணும்? It developed it to collect all the data which is available in all the literature, and based upon the literature, it collects the data which is the suitable ingredient for this particular drug. Example, suppose you are taking the paracetamol, so paracetamol வந்து lactose is good, and starch is a good detergent agent, and magnesium is a lubricant. So 5%, 15%, 12 to 30, 40% like that you have to use. So the information within short period of time the A will give. நீங்க the just data கொடுத்தீங்கன்னா போதும். paracetamol tablet பண்ணனும், அதுக்கு வந்து என்ன ingredient யூஸ் பண்ணனும், அது என்னென்ன ரேஞ்ச் யூஸ் பண்ணனும் நீங்க क्वेश्चन கேட்டீங்கன்னா the A will give the correct answer. How? It will be creating a lot of interaction between each and every molecule. X1, X2, X3, first they will interact each other with the paracetamol drug. It will find out, finally giving the optimize the output Y1, Y2. Y1 is the response. So paracetamol on the quick release within 10 minutes la it computer release ஆகணும். DT வந்து ரொம்ப ஃபாஸ்டா இருக்கணும். இந்த ரெண்டு the Y1, Y2 quick achieve பண்றது வந்து என்ன மாதிரி ingredient வேணும் நீங்க கொடுத்தீங்கன்னா it will give the correct results. புரியலையாப்பா? ஃபர்ஸ்ட் வந்து x1, x2, x3 ங்கிறது ஃபேக்டர். எந்த ஃபேக்டர் என்னென்ன ஃபேக்டர் வந்து உங்களுக்கு தேவைப்படுது அத ஒரு ப்ரிப்பரேஷன் ஆஃப் paracetamol tablet வந்து என்னென்ன ஃபேக்டர் தேவைப்படுது கொடுக்குறீங்க. என்ன பண்ணி என்ன பண்ணோம் எல்லா அந்த மூணு கொஸ்டின் எடுத்துக்கிட்டு it will interact each other, it will give the correct results based upon your input y1, y2. y1, y2 nothing but is a response. Response என்ன இருக்கணும்? paracetamol tablet should be dissolved and DD should be fast. இந்த ரெண்டு ரெஸ்பான்ஸ் அச்சீவ் பண்றது வந்து என்ன மாதிரி இன்கிரிடியன்ட் யூஸ் பண்ணுங்கிறது வந்து AN will be trying to give it this short period of time. இதுதான் பேசிக் கான்செப்ட். So first one is the input, அப்புறமேல தான் இது input node layer and hidden node layer, output node layer, finally gives the output. The node layer nothing but interaction between the factors. Multiple time interact each other, it will give the finally one output. புரியுதா? input and output முன்னாடி கண்டுபிடிச்சா நடக்கும்னா it interaction each other with each and every x1, x2, it interact each other, then x2, x3, then x1, x3, அப்புறம் x3, x2 அந்த மாதிரி மூணு மாறி varies level also. So நிறைய time interaction நடந்து finally gives one output, that output will be matched for the final product. So now people are using the preparation of nanoparticle. So if you want to prepare a nanoparticle, the same process. So now you have the data. You have data with literature. From the literature, you can collect the data. What type of nanoparticle you have to prepare. That data will enter into the ANN. First the input layer X1, X2, X3. Then it will interact each other. That is called hidden layer. Then finally it will give the output. That is called output layer. Then finally it will give the response whether it is produced nanoparticle or not. You can easily find. Without doing any lab work, by using the ANN, you can find out this material can prepare the nanoparticle or not. You can easily find. So one of the major advantages of artificial intelligence. புரியுதாப்பா? ஸோ நீங்கள் டெவலப் பண்ணாலும் சரி நானோபடி பண்ணாலும் சரி ஏஐ வச்சு நீங்கள் ஈஸியாக கண்டுபிடிச்சிடலாம். அதாவது பண்ண முடியுமா பண்ண முடியாதது வந்து fraction of second you can find the results. Next. So here you can see one example. So if you want to prepare a nanoparticle, you are given all the input time of iron oxide, cooling rate, temperature, எல்லாமே நீங்க ஒரு ஃபேக்டரா கொடுத்தீங்கன்னா it will interact each other, you will finally give the output for particular nanoparticles. So one more example, bioactive material using nano. So from the algae, you can find out the nanomaterials. So you can prepare by using the through AI. First you have to take the bioactive nanomaterial, then you can use the input layer, then it will interact each other from the hidden layer, then finally give it the output layer. That material will be tested into the animal model. You can finally find out this biomaterial can be useful or not. You can easily find it. Next. So listen, this is a very important slide. This note பண்ணிப்பா. இது நோட் பண்ணிக்கோ. அவ்வளவுதான். வேற ஒண்ணும் கிடையாது. AI வந்து எப்படி வர்க் ஆகுது அப்படிங்கறது பிரின்சிபல் தான். So AI சொல்றேனே அது எப்படி சார் வர்க் ஆகுது நீங்க சொல்றீங்க ஓகே இது எப்படி வர்க் ஆகுதுனா ஃபர்ஸ்ட் என்ன பண்ற ஃபர்ஸ்ட் process என்ன ஃபர்ஸ்ட் வந்து டேட்டா ஜெனரேட். Data generation from the literature. Literature எப்படி எடுப்பீங்க? ஜரா கூகுள்ல போட்டு பார்க்கலாம். இல்ல கவர்மெண்ட் இருக்கு, சயின்ஸ் இருக்கு, ஏதோ வெப்சைட் யூஸ் பண்ணி you collect the literature. என்ன மாதிரி ஒரு ப்ராப்ளம் இருக்கு யாரெல்லாம் எடுத்துருக்காங்க. So first structured and unstructured data. The data என்ன பண்றோம்? Once you put the AI, the AI will be arrange the data based upon the year, based upon the work, based upon the type of material. So the AI will be done first arrange the data. The big data will be arranged to the data and stored to database and servers. Then the AI will process the data. The based upon the process using the algorithm, you can give the output. So என்ன மாதிரி output வேணுமோ நீங்க கொடுத்தீங்கன்னா அது மாதிரி அது என்ன பண்ணும் AI will be create an algorithm. That algorithm வச்சு நீங்க வந்து சொல்லிக் கொடுப்பீங்க. Then finally you can give the correct output. What are the output you want? It will give the correct action of management. So first big data collection. Next one என்ன பண்ணுவோம்? The data will be aligned. The third one is applying algorithm. Final one is the assess the data. The நாலு process தான் நடக்கும் ஒரு artificial intelligence யூஸ் பண்ணி. சிம்பிளா சொல்லணும்னா ஒரே நாலே process தான். First collection of data, then arranging of the data and algorithm of data, then finally output of the data. This is what a best example. You want to develop one disposing tablet. Okay va? ஒரு disposing develop பண்ணனும். அத வந்து கொஞ்ச நேரம் இருக்கு. ஒரு disposing develop பண்ண போறீங்க. அத வந்து AI யூஸ் பண்ணி பண்றோம். புரியுதா? ஒரு எக்ஸாம்பிள் வந்து என்னது? Preparation of the disposing tablet by using AI. First என்ன பண்றோம்? So predicting the water disposing tablet formulation by neural network techniques. நோட் பண்ணிப்பா. சோ இதெல்லாம் எப்படி ஒரு வால் டிஸ்போசிட் டெவலப் பண்றது வந்து AI எப்படி யூஸ் பண்ணலாம் அப்படிங்கறது எக்ஸாம்பிள். So first, you have to first data extraction. First, you have to use the various websites, various machines, and find out collect all the data. From the data, you can go for all the search paper and certain paper extra data. Then you go for formulation and dissemination time. First, from the data, you have to fix the dissemination time because this war dissemination tablet. That's dissemination time வந்து fix பண்ணு. அதை fix பண்ணிட்டு அதுக்கப்புறம் என்ன பண்றாங்க எவ்வளவு டேட்டா கிடைச்சிருக்கு 140 டேட்டா கிடைச்சிருக்கு. இது பாருப்பா சைட் பாருங்க 145 nothing but from the AA you will get the 145 data based upon your input. நீங்க என்ன input கொடுத்தீங்க you want to develop the formulation of oral dissemination tablet. அதுக்கு எவ்வளவு டேட்டா வந்திருக்கு 145 டேட்டா வந்திருக்கு. அப்புறம் என்ன ஆகும் the 145 data வந்து the AA will be classified in 3 set. the AA என்ன பண்ணும் இந்த டேட்டா வந்து மூணு வேவா பிரிச்சுக்கும். ஒன்று வந்து ட்ரைனிங் ஆகும், இன்னொன்று வந்து வேலிடேஷன் ஆகும், இன்னொன்று டெஸ்டிங் ஆகும். எப்பவுமே அது ஒரு ப்ராசஸ் நீங்க புதுசாக டெவலப் பண்ணீங்கன்னா ஃபர்ஸ்ட் அந்த ப்ராசஸ் வந்து சரியா இருக்கா தப்பான்னு கண்டுபிடிக்கும். அதுக்காக என்ன பண்றோம் இருக்கிற டேட்டா வந்து the AI will be classified 3. One is the training, validation and testing. So அது நீங்க என்ன பண்ணீங்க ஃபர்ஸ்ட் வந்து அந்த AI மாடலை கொடுத்துட்டு ட்ரைன் பண்றீங்க. ஸோ இந்த டேட்டாவை வச்சு எனக்கு வந்து கரெக்டான ஒரு ரேஷியோ the part you have to give. So based upon that, the training and validation, you give the neural network, then you finally find one model. That model will give the precision accuracy. So first, you fix the decision time. Decision time will be less than 10 minutes. Last 5 minutes. So decision time for less than 10 seconds, which is suitable for my formulation. So the AI will be find out. So whatever combination you have to generate, that combination will give the final result, which is match your decision time. So if the model gives the exact results, so there was so decision tablet another decision the tablet has to be decision very fast. So when you வந்து எதுமே ஒரு எக்ஸ்டென்ட் ப்ரோக்ராம் அந்த எக்ஸ்டென்ட் வந்து நீங்க வந்து ஒரு டிடி டேபிளா மாத்தணும். நீங்க என்ன பண்ணுவீங்க you will go to the lab, you will take the drug and you take the inactive ingredient and mix it and compress it and finally get check the DT value. அந்த மாதிரி நீங்க எத்தனை டைல் எடுப்பீங்க டைல் எடுத்துட்டே இருக்கணும். உங்களுக்கு வந்து ஒரு 10 seconds வர வரைக்கும் என்ன பண்ணணும் number of time you have to take and find out the optimum competition. அதை நீங்க வந்து என்ன பண்ணலாம் by using the AI we can shorten the process without doing any lab work. We can optimize the formulation. That is the magic. By AI will be helpful collecting the data. From the data it will be analyze the what are the results already published. From that it will be developed a model. That model will be validated by you whether it's correct or not. After the once you have to perform the product in the lab, then you can finally find out the DD time. The DD time matching the AI data, the model is valid. If suppose the observed value is not matching the AI data, the model is not valid, then you have to change the other model. Clearly, DD வந்து இது ஒரு ஆப்டிமல் காம்பினேஷன் கொடுக்குது இல்லையா கம்போசிஷன் கொடுக்கும். அந்த கம்போசிஷனை வந்து நீங்க வந்து லேப்ல பர்ஃபார்ம் பண்ணி பார்த்துட்டு அந்த வேல்யூ ஈக்குவலா வந்துச்சுன்னா அந்த மாடல் இஸ் வேலிட். சப்போஸ் அந்த மாடல் வந்து நீங்க ஃபாலோ பண்ண மாடல் அதை நீங்க அப்சர்வ் பண்ணி லேப்ல பண்ணும்பொழுது அந்த மாடல் அதே வேல்யூ வரல அப்படின்னா that model is wrong, you have to change the model. புரியுதா அந்த மாதிரி நீங்க என்ன பண்ணணும் by using the AI we can take the single file for a single file you optimize the formulation, not necessary to go for the headboard trials. Next. So listen here we are taking the two network. One is the ANN, other one is DNN. ANN is a very familiar one and DNN is the most இன்னொரு ரொம்ப அதிகமானது வந்து DNN. So ANN is the artificial neural network, DNN is a deep neural network. So there are multiple times the interaction will happen at DNN. ANN ல வந்து one time வந்துச்சுன்னா DNN ல வந்து 10 times நடக்கும். The interaction between the layer ANN ல வந்து single time நடந்துச்சுன்னா DNN ல வந்து இருக்கும் 10 times நடக்கும். So ANN is a single process and DNN is a multiple step process. How to think about ANN is the single step process, DNN is the multi-step process. So So AN is basically blocked with the interconnected layer, but the DN is the deepest of the AN multiple. So hidden layers of the upper hidden layers will be keep on increasing the DNL layer. So here you can see, first you give input, first the APA and the molecular descriptor. So the APA is the drug molecule, the drug molecule is molecular input. First input. Second input, excipient. Whenever excipient you are going to use it, the excipient coding good to the how much quantity of APA and the excipient you are using. The moon value on the AI input drug characterization and excipient characterization and quantity. The moon data moon input AI machine. So the machine it will be intact each other and finally give the what DD time. So this is the combination you can use it based on you will get the this is the DD time you will get. Next slide. So now here you can see the DNL. AN will be the single time interaction, but DNL you can see 4 times it interact each other. So multiple times. That's a multi-step process. So all the input will be hidden like from the hidden layer, it interact each other, it gives the finally one single output. So then the result value will be very accurate one. The number of times we interact each other, so the final value will be the accuracy. Next. So this is the result now. You can see for paracetamol drug, for API, so the results will give in. First you have to take the 224.4 mg of paracetamol. The next one is the what is the full filler you have used? Mannitol. What is the quantity? 3.6. These are based upon literature data, not your lab data. Based upon the literature data, it will give the this is the result. So for APA, this is the quantity you have to take. For filler, this filler is good. This is the quantity you have to take. The binder, this is the binder is good. This quantity you have to take. Binder is a start. 25 milligrams is the quantity. The disintegrant, Sastha sodium, the quantity is 44.4. Lubricant, magnesium, dose is the 3. Solvent is the SLS, sodium sulfate, quantity is the 8.6. If the input what type of output will get? This is the tablet output. One is the hardness and variability, thickness, pulse and the DD time. So this is the output. This is the lab. So whatever the input you are giving, the based upon the interaction, it will give the final output. So if you are taking this combination in the lab, you are performing the practical, then you will get the results. The variability, hardness, thickness, and daily time will be over. So this is all match all is valid. It is not match, all is not valid, then you have to change the model. Very simple. So you have computer AI open input the output. The output you take only one trial. What same result the model is valid. The result will be vary, the model is wrong. Next. So here you can see the formulated with mannitol, formulated with non-mannitol. So with mannitol. So this gives the different combinations. So first one is the paracetamol, this is the formulated. Next. This is a very important slide. So if you want anyone to try for the artificial intelligence, just go to the website. If you just go to the website and try it. If you want to try it, you will get some idea. So for neural network, you can use deep learning 4G.org. So you try the predictive model. All the sources can be found in the website. Why.net for pharmaceutics will get all the idea. Just to go this website and try it. If you have any doubt, you just contact me. slide back. Listen here. So for BB tablet, as per the European pharmacopoeia, the OA tablet is a disintegrated within 10 minutes in the mouth before entering into the stomach. These are one of the limits. So if the limits are set, the AA will be find out which of the composition will come to the particular results. That combination will be only highlighted in your results. As per the European pharmacopoeia, the OD tablet, the word disintegrated tablet is called as it should be disintegrated within 10 minutes in the mouth before it's swallowed. So the முன்னாடி 3 நிமிஷம் தான் வாயில கலந்துரும். அதான் வந்து ஓரல் டிசின்டெகிரேட்டட் டேப்லெட். புரியுதா? சோ அந்த மாதிரி ஒரு ப்ராடக்ட் வேணும் அப்படினா நீங்க ஒரு இன்புட் கொடுத்தீங்கன்னா அத AI வந்து will give the results. The based upon the results you will collect the combinations and you can work out it. You will get the correct results. The model is valid. If you are not getting correct results, the model is not valid. Then you have to change the other model. Okay. So listen, till now who are not understood, at least by seeing this slide, we will get some idea. So AI வந்து எப்படி யூஸ் ஆகும் அப்படிங்கிறது வந்து இப்போதைக்கு புரிஞ்சுக்கணும். இவ்வளோ நேரம் ஒன்றும் புரியல அப்படின்னா கூட பரவாயில்ல. ஓகே லீவு. இந்த சைடை பாருங்க, உங்களுக்கு ஈஸியாக புரியும். எல்லாம் நம்ம டெய்லி யூஸ் பண்ணுறது. இப்போ வந்து என்ன பண்ணீங்கன்னா AI வந்து யூஸ் பண்ணி we are finding the blood pressure of the patient. புரியுதா? ஸோ ஒன்று என்ன பண்ணீங்கன்னா so far இப்போ நீங்க வந்து சிம்பிளா வந்து டாக்டர்ட்ட போய் பிபி மட்டும் பாருங்கன்னா டாக்டர் என்ன பண்ணுவாரு ஜஸ்ட் என்ன பண்ணுவாரு அந்த பிபி பாட்டஸ் வந்து கண்டிப்பா பிபி பாப்பாரு but the BP will vary the various other factors. The doctor will check the BP in the patient, but the doctor may not consider for the all other factors. புரியுதா? உங்களுக்கு சிச்சுவேஷன் நான் ஸ்ட்ரெஸ்ஸா இருக்கீங்களா பிஎம்ஐ இருக்கீங்களா இல்ல எல்லாத்தையுமே செக் பண்ண மாட்டாங்க டைம் ஒரு வாட்டி நீங்க போய் டாக்டர் 1000 அப்படின்னா அவர் வந்து பிபி செக் பண்ணுவார். பிபி செக் பண்ணிட்டு பிபி அதிகமாக இருக்கு கம்மியாக இருக்கு மட்டும் சொல்லுவார். அது மாத்திரை கொடுப்பார். பட் அந்த பிபி ஏன் அதிகமாக இருக்கு ஏன் கம்மியாகுது அப்படிங்கிறதெல்லாம் சொல்ல மாட்டார். அதுக்கு வந்து you have to go for some other test. But if you are having the AI in your instrument, BP instrument, it will take all the input. You are giving the one other input, lifestyle cooking. Lifestyle is the activity, what activity you are doing, what type of meal you are taking, what is your BMI, and what are other lifestyle. Either you have smoking habit or not or alcohol. So what are the habit you have, you can just give the input. இது எல்லாமே நீங்க கொடுக்கலாம் ஒரே இது. அப்புறம் என்ன பண்ணீங்க என்வாயர்மெண்ட் வந்து ஸ்ட்ரெஸ்ஸா இருக்கீங்களா இல்ல டெம்பரேச்சர் அதிகமா இருக்கா அப்படி. அப்புறம் ஜீனோம் 30 உங்களுக்கு பிபி இருக்கா இல்ல ஃபேமிலி ஹிஸ்டரி ஏதா இருக்கா ஜீனோம் இது எல்லா ஃபேக்டர் என்ன பண்ணலாம். We will give us the input of the AI. The AI will be analyze everything. It will give the final what is your average BP in your body. Any cases. புரியுதுலப்பா டாக்டர் நம்ம போய் பிபி செக் பண்ணும்போது டாக்டர் போய் பார்த்தோம்னா அவர் மிஷினை வச்சு என்ன பண்ணுவாரு will find out the BP at person what is the based upon that will find out whether the BP is high or not. Based upon that will be the tablet. But doctor can analyze all other factors. But AI can analyze all the factors which is the cause to reduce the BP in your body. Either food habit ஆகுதா இல்ல உங்க லைஃப் ஸ்டைலா வருதா இல்ல என்விரான்மெண்டா வந்ததா இல்ல ஹெரிடிட்டியா சோ எல்லாத்தையும் என்ன பண்ணலாம் சிங்கிள் டெஸ்ட் you can find out all the reason causes for the BP. அதான் main major advantage of AI. So single for fraction of second you can find out the proper cause of the diseases. புரியுதா? புரியலையா? So AI நீங்க யூஸ் பண்ணீங்கன்னா you will find out the lot of other various factors, but time is very short. Very short period of time you can find out the results and root cause of the disease. So these are some tools of the AI. If only you can note it down. Whenever you have the free, you can just search the Google. You can find out a lot of other tools, AI tools is there. You just apply in your research. While doing the BCom project work, you just try to apply once you get some idea. So now conclusion slides. So already told AI has become the game changer in the pharmaceutical industry. What is game changer? It will be completely another computer change process வந்து 6 months within a month of process முடிச்சோம். So game changer in the farms industry. The drug formulations developed require extensive testing on the optimized variable parameter, but AI model can quickly analyze the data of multiple identify the most promising formulation. So நிறைய டைம் நீங்க வந்து நிறைய டைம் கையில் எடுக்கணும் அவசியம் இருக்காது. So you can completely avoid the times. You can using the AI, you can optimize the formulations. Last, so due to that, you can reduce the cost of the company and time, and you can save the manpower. So these are all very useful for in farms industry by getting by the AI. Okay, thank you. Any doubt?
Thank you, sir. It was very informative. So I just have 2 questions to you. So one was, you said that in the BP pharmacopoeia, it has been told that totally is disintegrating in 3 minutes. But AI has basically tried to understand that. Can you clarify that?
If you want to, that is based upon the European pharmacopoeia, we have 3 steps. So for this, we have the research. Now you have the age drive. The age drive should be designed with 3 steps. So you know what type of composition.
Guys, please be seated.
Based on the
Guys, please be seated and remain silent.
Blood pressure வந்து இது சொன்னீங்க. So என்னோட question என்னன்னா, நீங்க என்ன சொன்னீங்க, நம்ம டாக்டர் வந்து பார்க்கும்போது he will just tell what is the symptom. But AI ல வந்து have too much of information. But this one question, will it be specific to that person, or is it just going to give everything, like whatever information? AI is like database stored, yeah?
If suppose if you want to compare the BP of the particular area in Chennai, due to the temperature or due to stress or what the people, if you want to go on, Chennai or the population around 1 crore, so 1 crore will be not taken. You can write only for you can analyze what is going on in this mind. Whether they also have the BP, what is the impact of the BP. That can be possible in AI. So don't need to go for all the population and take the whole time. You can easily manage the model by using the AI.
Very interesting future way is going to handle it. And I have one doubt, sir. If you have a diabetes materials, right? So which type of diabetes materials can it consume, sir?
The reason is the diabetes component will vary to specific diseases, country to country, food, lifestyle, management. There are a lot of other factors involved. So if you want clearly identify treatment of diabetes factors, you have to treat the patient with all other factors. Not only the drug is not going to solve the problem, the patient has to change the lifestyle and food state and management of disease like that is different. So the AI will give the accurate whatever drug you have to take and what is the time and what type of food you have to take and what is the time of the food state and what type of lifestyle, whether you have to go to exercise or not, or avoid the smoking, avoid the alcohol. So எல்லாமே ஒரு history மாதிரி data create பண்ணி not only giving only the tablet. Doctor can only give the tablet prescribe the drug and the quantity of the symptoms now, but they will be analyzed all the other factors of the body. What it is the correct result. So this is the drug, this is the lifestyle, this is the food, this is all the data. That's what the AI will do to analyze the data and give the result justice and compare to human manpower.
Thank you, sir, for your wonderful and innovative session. Hope your session has enlightened creative ideas from the mind of young delegates present here. As we come to an end of today's part of conference, the success of this event and the direction of the conversation have been greatly aided by our senior staff, Mr. Mubarak. It's my honor to invite you, ma'am, for delivering the closing remark. Over to you, ma'am. Thank you, ma'am.
A very good evening to eminent speaker Dr. Ubaidullah sir, the faculty, guest faculties and all the students presented over here. Now it's the time to be and end this evening to start a wonderful tomorrow's morning. So if we look at an overview of today, we have two wonderful sessions. In the morning session, we started with our principal sir speech, which was followed by Dr. Balaji Ramachandran sir's topic on in vivo imaging as a non-invasive screening tool in translation research, which is very helpful for the biogenetic engineering students and pharmacological students in translational research who were interested in translational research. So which was simultaneously we are conducting oral presentation at the pharmacy block. So all the students participated enthusiastically. So while talking about the commencement of the afternoon session, we started our session with our chairman sir's speech, which was followed by Dr. Ubaidullah sir's session on the topic AI's prominent directive towards designing EDS and PT. I hope all the students got awareness on the topics. It's the time to convey my thanks on behalf of our pharmacy institutions to the chairman sir and principal sir for permitting to conduct the conference in the institution and for continuous guidance throughout the session respectively. Secondly, I thank to our speakers who extract their time from their busy schedules and came here to enlighten our knowledge. So I thank to all the committees including stage, food, discipline. So without their cooperation it is impossible for the successful completion. Last but not the least, I thank to all the participants for all their bearing patience. Thank you.
We are grateful for you, ma'am. Thank you to all of you for your participation and valuable contribution for the day. Please collect your refreshment while going out.